Hello. Hello, am I audible?
Yes, ma'am.
Yes, ma'am.
Okay.
Yes, ma'am.
So a very good morning to all the faculty members of different academic institutions present here in the online refresher course. Our speaker also joined. Good morning, sir.
Yes, good morning.
Okay, sir. So may I start?
Yes,
So it's
please.
my it's my pleasure to introduce today's resource person, Dr. P. Gopinath, professor in the Department of Biosciences and Biotechnology in the Indian Institute of Technology, Roorkee. He earned his PhD in biotechnology from the IIT Guwahati. He did his postdoctoral work uh, at the University of Rochester Medical Center, New York. Today, Sir Dr. Gopinath will deliver a lecture on nanotechnology on cancer diagnosis and therapy, a topic that remains one of the most significant scientific, medical, and social challenges of our time. Um, Despite how common the term, cancer is not a single disease. It is a complex family of more than 200 distinct disorders, each with its own biology, behavior, and responses to therapies. Cancer begins when healthy and normal cells lose the ability to regulate growth, mutations accumulate, and driving cells to divide uncontrollably, evade the immune systems, and sometimes spread to distant organs by metastasis. These changes arise from a combination of factors: inherited and genetic predisposition, environmental exposures, lifestyle influences, and random cellular events. So, our speakers will uh, enlighten us on these very current topics on nanotechnology on cancer diagnosis and therapy, and his research group in the nanotechnology laboratory at IIT Roorkee is working on developing various nanomaterials for biomedical applications. At present, he has more than 170 research publications in the area of nanobiotechnology in high impact factor journals. He has filed 30 patents, out of which 17 granted, and done one technology transfer. He has also published 8 books and 15 book chapters. His research work has been well cited with total citations of more than 9,620, high index 54 and I-10 index 135 till date. He has completed 15 sponsored research projects so far and currently working on 10 projects sponsored by various funding agencies like ANRF, DBT, DST, ICMR, etc. Further, he has received several prestigious awards, including ILS1, India Young Scientist Award 2021, Tata Innovation Fellowship 2024 by Department of Biotechnology, Government of India. He is a Fellow of Royal Society of Chemistry, FRSC, and Fellow of Royal Society of Biology, FRSB, United Kingdom. He is the co-founder and director of startup company Super Good Nano Private Limited. So, with this introduction, may I request sir to deliver your lectures, and my humble request to all the participants here, if you have any questions, you can put your questions, but our due to shortening of time, I will request you just to raise only one questions, that too in the discussion portion of this lecture, that will last for last 10 minutes. Thank you, sir. Thank you, everyone. Please go ahead.
Yes, thank you. So I'm audible, right?
Yes,
Okay, I'll just share my slides so that. So my screen is visible.
yes,
Okay, the slides are moving, right?
yes.
Okay, okay, fine. So, very good morning, everyone. So, thanks for the nice introduction and also the wonderful opportunity to interact with all the faculty colleagues. So, today I'll be talking about the nanomaterials for cancer diagnosis and therapy. So, in my talk, initially I'll talk about what is nanoparticles and how to make nanoparticles. Then, followed by that, I'll talk about briefly on what is cancer. Then, I'll also talk about like how nanomaterials help in cancer diagnosis and cancer therapy. And at the last, I'll also show you some of the research work which we are doing in our lab. So it will give you a broad idea about this particular topic, how nanomaterials play important role in cancer diagnosis and cancer therapy. So let me briefly start with what is nanoparticle. So nanoparticle means any material having at least one of its dimensions should be in the range of 1 to 100 nanometer. So whenever you take any material, so it will have x, y, and z, right? So out of that, at least any one of its dimensions should be in the range of 1 to 100 nanometer. Then we can call that material as a nanomaterial. And most of you know the word nano is a Greek word that means it's small. And again, the most of you know the nanometer means it's 10 power minus 9 meter. So to make it more simpler, uh, we can compare the total population of India. The total population of India is 100 crores, which is 10 power 9. So when you compare the yourself to the whole Indian population, you and me are kind of nano size. Okay, so that much small is the nano. And one of my relative also asked me like, you are working in nanotechnology, can you tell what is nano? So I just told him a simple example, which is equal to 1 rupee in 100 crore 1 rupee coin. So that much small is the nano. And why small is good? What is the advantage of this small particle, nanoparticle? If you see that whenever the size goes down, the surface area to volume ratio will be higher. For example, this is a 7 shell, the surface atom is only 35. So whereas this is a 1 shell, and you can see the surface atom is 92. When the material is small, so which will have various remarkable properties, like for example, it will be cheaper, it can be efficient, and it can perform some new functions. For example, the gold, which is the inert material, so when it goes to the nanoscale, it can be useful for cancer diagnosis and cancer therapy. So how the surface area to volume ratio will be higher? So we can see this simple example. You take a 2 box and you are cutting the 2 box into 1 So this is like a kind of bulk material, and this bulk material you are converting into nanomaterial. So when you see this uh, same 2 box, when you cut into 1 box and apply the surface area formula, you can see that the surface area is increased, but the volume is same. So the surface area to volume ratio will be higher when the size goes down. So that's why when you make from bulk to nanomaterial, the surface area to volume ratio will be always higher. And additionally, the advantages is the hidden surfaces are exposed, so which can have a better catalytic activity. The simple example for understanding this concept is maybe you can purchase the aspirin tablet or you can purchase the Eno tablet. So just put in the water, it will take 50 seconds to dissolve. The same, you break it into small powder and put in the water. Within 10 seconds, it will dissolve. So that is the advantage. When the size goes down, it can have a better catalytic activity, and also all the hidden surfaces are exposed. And so that is the reason. So in the bulk to nano, which will have a more surface to volume ratio, and it can have a better catalytic activity. Another simple layman example is when you put a chalk piece in the water, it takes more time to dissolve. The same chalk piece, you break it into powder and put in the water, it dissolves very quickly. So that is the difference. And similar to the biotechnology, this nanotechnology is also not a new field. So it was there for several years back. So, for example, those days they use this kind of beautiful paintings on the church windows, and those days they don't have any branded color paints, but they had more amount of gold and silver. And when you're using this gold and silver of different size, and when you make this kind of painting with respect to the transmitted light and with respect to the reflected light, you'll have a different kind of colors. So, for example, this is a Lycurgus cup, which was used in the 4th century by the Roman kings, and you can see that it was coated with the gold and silver. And with respect to the transmitted light, it will give red color, and scattered light, it will give a green color. So those days don't have the LED or some other light. So, but they had a lot of gold and silver. Using this, they are played with the this kind of colors. And again, it is not new to the our Indian traditional medicines. Like for example, some of the basmas, it has the gold and again silver for the various therapeutic applications. So, how to make the nanostructures? There are broadly two approaches: top down and bottom up. So, the top down is like making a statue from the rock. So, this is your bulk material. So, from the bulk, you are making the nano material. So, from a very, very big size, you are reaching the nano size, top to down. So, that's why it is called as top down approach. And next one is bottom up approach. So, bottom up approach is from a very, very small size, you are making the nano size. So, for example, using the brick and you are constructing the building. So here, for example, most of the biological nanomaterials comes under the bottom-up approach category. So, for example, if we have a protein, so the protein is made up of amino acids. So, amino acids are the building blocks for the protein. So here, these amino acids are like the brick. So this size of each amino acid is in the range of angstrom. So these amino acids combines and forms the nano size protein. So this is called bottom-up approach. And same thing with the nucleotides, ATGC. Okay, so these also in the range of angstrom. So this combines and forms the nano size DNA. So this is called bottom-up approach. From a very, very small size, you are reaching to the nano size. And when you talk about the nanoparticles, so the size and shape, so everything plays a very important role. So we can make a different shape and we can also make a different size. And we can also use any kind of material, like it can be a polymer or it can be the polymer plus lipid conjugation combinations. So that is also possible. So very important thing is the surface charge. So whenever you make a nanoparticle, so most of you know the charge of DNA and the cells are negative. Okay, so when you want to have an antibacterial nanomaterial, most of you know the charge of bacteria or the cell is negative. And when you have a positive charge nanoparticle, so which can easily come and bind to the bacteria or cell or DNA through the electrostatic interaction. Or what we can do is we can also add a specific antibody or peptide which will go and bind only to the cancer cell. So that I'll talk detail later. So let us see what is the difference between nanoscience and nanotechnology. If you see that nanoscience means if you're making a fluorescent nanoparticle and your BTech students or MSc students are making some publication or some thesis report, so that's the science, and you are using the same fluorescent nanoparticle and you are making some product, some technology, then it's called as nanotechnology. So what is the difference between nanobiotechnology and bio-nanotechnology? So most of the times these terms are used vice versa, but ideally, nanobiotechnology means the applications of nanomaterials in biotechnology, whereas bio-nanotechnology means understanding the biological nanostructures. So biological nanostructures, for example, DNA and protein, if you're able to understand these biological nanostructures for various applications, so that is called bio-nanotechnology. This is the biological nanomaterials. So we got the overall idea about what is nanomaterials. So now we'll briefly go on understand how to make these nanoparticles by physical method, chemical method, and biological method. So there are several techniques in the physical methods. I'll briefly talk about two important techniques. The first one is mechanical milling. It is also called as ball milling. So here we'll be using this kind of balls. So these balls can be polymer or it can be metal. So it depends on the final applications and depends on the availability of the jar and the balls. Okay. So when you put the the balls in this container, the jar, so what will happen is these balls will collide and it will make your micro size particle into the nano size particle. So it has attrition and impact. So due to that, it will grind your powder into nano size. So it is similar to the mixer grinder. Mixer grinder will be using the sharp blades. So, but it cannot reach to the nano. But after you grind in the mixer grinder, then you can use that powder and put it in the ball milling. So it keep on grind and makes to the nano size. So this is how the equipment look like. So this is a jar and this is the balls. And you can see that the figure A, so after 3 hours, the size is bigger. And figure D, you can see this. After 20 hours, the size gone down. So if keep on milling, the size will go to the nanoscale. Depends on the final, this your powder or depends on the material and depends on the jar and balls which you select. So another simple technique is laser ablation. So we can use the laser to vaporize the material. For example, during your childhood, you might have applied this laser light on the cotton or the tissue paper. It might have burnt. Similarly, instead of that, we'll be applying the laser light on the your gold or the particular silver, which nanoparticle you want to make it. So this is also a kind of eco-friendly and environmental friendly method. It's a green method because we are not using any toxic chemicals for preparing these nanoparticles. And if you see that, so these nanoparticles are directly made from the metals, so it will be very useful for biological and in vivo applications. So we'll be using the laser called ND-YAG laser, and we have to keep the particular metal target. So if you want to make a copper nanoparticle, you can keep the copper slice. If you want to make the gold nanoparticle, you can keep the thin slice of gold. And using this XYZ stage, we can move this and make this complete metal into vaporize into nanoparticle. So this is your metal target. It can be gold or silver or copper. Apply the laser light and you can collect in the aqueous media or organic solvent, depends on your final application. So the nanoparticle can be collected. And the next method is chemical method. So in the chemical method, for making nanoparticle, we need three important components. The first one is metal salt. So chemical method, I will be focusing only on the metal nanoparticle. There are several nanoparticles like polymer and other materials. I'll try to discuss later. So if you want to make a metal nanoparticle, for example, you want to make a silver or if you want to make the gold nanoparticle, so you can use the particular metal salt. Then you have to select the correct reducing agent. So this reducing agent will reduce your metal salt into metal nanoparticle. But this metal nanoparticle is highly unstable. So to make it stable, we have to add a capping agent or a stabilizing agent. Okay. So the stabilizing agent or capping agent, it can be acetate or it can be the polymers, so which will provide the stability to the nanoparticle. So for example, as I told you, this is the bulk gold, which is a shiny yellow metal. So when it goes to the nanoscale, you can see the color and properties will get changed. So the reason for this color is surface plasmon resonance, that is called SPR. So that is a surface plasmon resonance, mainly for the metal nanoparticle. When the size goes down, it shows some remarkable colors. And what is SPR, surface plasmon resonance? So most of you might have seen this whenever you go to the jewelry shop. You'll have shiny metal, and its shining is due to the electron cloud. Whenever there is a light, the metals are shining it due to the electron cloud. And these electrons have dual nature. When you're having this light of matching wavelength, it will produce a resonance. So for nanoparticles, this wavelength lies in the visible region. So that's what you're getting these beautiful colors. So when you apply the light, there will be oscillation of conduction electrons. So that leads to the surface plasmon resonance. So with respect to the size, with respect to the shape, the color will change. There will be a red shift with respect to the increase in the particle size. So when the particle size is bigger, so the peak will be like shifted towards the red region. So how these nanoparticles are formed, there are two important steps in the nanoparticle, that is the nucleation and growth. For example, any biological cell, how the nucleus is important. Similarly, for nanoparticle also, the nucleation is very, very important. So the metal salt will be reduced into the metal atoms. So these metal atoms combines and form the small clusters, and it forms the small nanocrystal. That step is called as nucleation. So the formation of the first nanocrystal in the solution is called as nucleation, and followed by that, these crystals will try to combine and start growing into a smaller size particle, nanoparticle. We have to control the growth by adding the capping agent. Otherwise, it will keep on grow, and which leads to the bigger size particle. That means the nanoparticle is aggregated, which is not useful for us. So we have to control the growth by adding capping agent or stabilizing agent, so which will prevent the particle growth and control the growth. So how we can control the growth by adding the stabilizing agent, and the stabilizing agent, there are two types of stabilization. The first one is electrostatic. So that means it will give uniform charge to the particle, and most of you know the uniform charge repel each other. So whenever you have a nanoparticles of uniform charge, so it will repel each other. And another method is steric stabilization. In the steric stabilization, so you'll be adding these polymers or surfactants, and which will provide a sterically bulky, and it won't allow the nanoparticles to come closer, or it won't aggregate. And you can see that the gold nanoparticle, usually the gold nanoparticle will be like a ruby red color, and if the nanoparticle is agglomerated or aggregated, it become like a purple or blue color. And the nanoparticle, when it's dispersed, it gives a sharp peak around 500 or 550 nanometer. And if they have a nanoparticle, aggregated nanoparticle, so you can see that it is providing the broad peak, and also the peak is shifted towards the red, and you can see that it's coming around 650 or close to the 700 nanometer. And whenever you make nanoparticle, the first step, metal nanoparticle, usually you see the color change. Whenever you see the color change, that means the nanoparticle is formed. In case of gold nanoparticle, it will be red color. In case of silver nanoparticle, it will be yellow color. And then you have to check the stability of the nanoparticle. So to check the stability of the nanoparticle, we have to do the zeta potential. So the zeta potential of the particle is the overall charge that the particle acquires in a particular medium. And the cutoff value is plus or minus 30 millivolt. That means if you're having a nanoparticle zeta potential above plus 30 or above minus 30, that means the particle is highly stable. So the same machine can also be useful for measuring the hydrodynamic size. That is the approximate size of the nanoparticle can also be measured using this same zeta sizer machine. So this is a zeta sizer machine, and so you have to press this switch. This is it has a laser as a light source, and so once it becomes green color, the machine is ready. You just press this button, it will open, and for measuring the zeta potential, we have to use this kind of cuvette, which is like a Y shape. So this is a small lid. We have to remove the lid, and we have to load this gold nanoparticle without any bubbles. And you can see that there is a small electrodes also available. So it will measure the charge of the nanoparticle, and the same machine can also be useful for measuring the hydrodynamic size, that is based on the dynamic light scattering. So here the cuvette will be normal plastic cuvette for measuring the DLS, and whenever there is a particle in the suspension, what will happen? The particles will be slowly moving due to the Brownian motion of the particles. So that will scatter the laser light. So that scattering can be measured, and it will measure the hydrodynamic diameter of your nanoparticle. That means, so for example, you are having a nanoparticle along with your stabilizing agent. So it will measure the complete size of the particular nanoparticle. And whereas in the transmission electron microscope, it will measure the exact size of your nanoparticle. A simple layman example is this is like that DLS is measuring the size of the fresh grapes. So the fresh grapes which has the water inside, so and so it will be like the size is bigger, and this TM is like a kind of measuring the size of dried grapes. So it is like exact size of the particular grape. So here nanoparticles along with the particular stabilizing agent, you will get it in the DLS, and whereas in the TM you get the exact size of the nanoparticle, because TM will be using the completely dried sample under the vacuum. And next one is zeta potential. What is zeta potential? As I told you, it gives the overall charge of the particular particle. So this is a nanoparticle, and some amount of charge will be traveling along with the nanoparticle. So that particular cutoff is called as zeta potential. So the electrical potential that exists at the slipping plane, this is called slipping plane. So that is called zeta potential. The simple example is when you put a stone on the pond, so it will give a like a ring structure, right? And the slowly the ring will diffuse. So along with the stone, how much the ring is very thick intensity. So that is a kind of zeta potential. So along with the nanoparticle, so the amount of charges which is tightly attached to the nanoparticle, so that boundary is the slipping plane is called as zeta potential. So beyond that, you can see that the charges are diffused. Okay. So if the nanoparticle is plus or minus 30 millivolt, that means your nanoparticle is highly stable. So you can see here, if we have this more charges, then it will repel each other. And if we have a low zeta potential, the particles will come closer and it will leads to the aggregation of the nanoparticle. And the basic principle of electron microscope, most of you know, the scanning electron microscope, which will kind of scanning. And simple example is like it's like your scanner. So it will scan the particular the question paper or the particular image. So in the transmission electron microscope, this is like your photocopy machine. So here the electrons are transmitted through your sample. That's why it is called as transmission electron microscope. And here it is scanning. So it is called as scanning electron microscope. And we can also make the silver nanoparticle similar to the gold. So here we are going to use the silver nitrate as the metal salt and sodium borohydride as a reducing agent. So this reducing agent will reduce the metal salt into metal nanoparticle, and this can also act as a kind of stabilizing agent. So when you make the silver nanoparticle will be yellow color. Gold is ruby red color, silver nanoparticle is yellow color. And once you see the yellow color, you are sure that you made a silver nanoparticle. And as I told you, so we have to measure it using the spectroscopy. When you use a UV visible spectroscopy, for silver nanoparticle, you get peak around 400. And you can also see that the ratio between the reducing agent and metal salt is very important. If the ratio is 2, it is stable. Ratio is 2.1, stable for 30 minutes. Ratio is 1.8, it's stable only for 5 minutes. So we have to optimize the right concentration of metal salt and reducing agent. So that ratio is very, very important. And based on the peak and based on the literature, we can also get the approximate size of your nanoparticle. If you're getting peak around 420 nanometer, the size of nanoparticle is approximately 35 to 50 nanometer. And final confirmation of the size and shape can be done by using the transmission electron microscope. And whenever you take TEM image, the scale bar is very important. So with the help of scale bar also, we can measure the size of your particular nanoparticle. And for the nanoparticle, you get the single peak. And for nano rods, you get 2 peaks. So one is for the transverse and one is for the longitudinal. So for nano rods, you get 1 peak for this and 1 peak for this. So you get the 2 peaks for the nano rods. So this is a simple protocol which we optimized in our lab. So these slides I already shared with the coordinators. So they will be sharing the slides. So you can simply make this nanoparticle by simply following this protocol and shake it. So you get the complete nanoparticle. So we got the idea of how to make nanoparticle by physical method and chemical method. So now let us see how we can use the biological system for making the nanoparticle. So for example, we can use these microbes and other plant tissues for making this nanoparticle. So in the chemical method, we have learned that we need 3 important components: metal salt, reducing agent, and stabilizing agent. So similarly, here also we need a metal salt. And instead of using the reducing agent and stabilizing agent, we'll be using this the microbes. It can be bacteria, or it can be yeast or fungi. Or we can also use the plant leaf or plant flower or the roots or stem, which has the phytochemicals. So, and this microbes has the enzymes. So this will act as a reducing agent, and it will also act as a kind of stabilizing agent. Similarly, phytochemicals, which can act as a reducing agent, and this can also act as a kind of stabilizing or capping agent. So the metal salt is reduced by this particular enzymes or the phytochemicals, and the final nanoparticle is ready for the end use. So the advantage of biological method is it's a eco-friendly method and low cost, and by simply changing the pH and temperature, we can get the different type of nanoparticle. So additional advantages like we can also get the synergistic effect. For example, you are making a metal nanoparticle which has a antibacterial, right? And if you're using the, for example, the turmeric plant extract, which also has the antibacterial, or if you're using the neem extract, which also has the antibacterial. So what happens is when you're making nanoparticle using this neem extract or turmeric extract, so that will also have some antibacterial effect. So it will have a synergistic antibacterial or anti-cancer effect. That is the advantage of this biological system. And not only the plant extract and microbial extract, we can also use the virus for making the nanoparticles. So for example, if you are taking a virus, and the main important component of virus is the nucleic acid. And when you remove this nucleic acid, what will happen is like you'll have only the particular small protein box, that is the protein container. And everyone know the size of virus are in the range of nanometer. So it can be a kind of small nano container for making your nanoparticle or loading your anti-cancer drug. So in this picture, you can clearly understand. So this is a virus protein cage architecture. So we can compare this the viral structure to the football. So you can compare it to the football structure, and you know that football, it has the outside interior, exterior, and subunit interface. So inside we have the football bladder where we fill the air. So that is the nucleic acid. We can remove the nucleic acid and we can put the anti-cancer drug. And the subunit interface, the hexagonal small leather piece of football is stitched. Okay, this is like a kind of stitching in the virus also. So the subunit interface where we can add the metal called gadolinium, so which will be useful for cancer diagnosis. And we can make it specifically this protein viral nanoparticle for cancer by adding the specific targeting ligand. For example, we can also use this 3 amino acid peptide, that is RGD, that is argine, glycine, and aspartic acid. So most of the cancer cells overexpress these receptors, so it can go and specifically bind to the cancer. And we can say this is a kind of multifunctional nanoparticle or a theranostic nanoparticle. So the theranostic means the single nanoparticle which can do the therapy and diagnostic. So that is called a theranostic nanoparticle. So you can see that inside you have the therapeutic molecule, and we have the gadolinium for cancer diagnosis, that is in the subunit interface. And outside we have this RGD, so which will make this nanoparticle to specifically go and bind to the cancer cell. And the advantage of using the plant virus when compared to animal virus is like plant virus are easy to produce, and it is available in different shapes and different size, and it is less immunogenic. So these are the advantages of this plant virus. And also it has a high loading capacity. We can load more amount of anti-cancer drug or therapeutic material. So that is the advantage of this plant virus. Okay, so easy to produce. So we are avoiding human virus to reduce the infections, and we can load more amount of anti-cancer drugs, and very easy to produce this plant virus. So this is how we make the plant virus. We can add the plant virus to the plant leaf. It can infect the leaf and make more number of plant virus. By doing the chemical treatment, we can remove the nucleic acid, chemical and enzyme treatment, we can remove the nucleic acid, and we can load our anti-cancer drug and study the efficiency of this nanoparticle, viral nanoparticle, on the your cancer cell lines. Then you can use the mouse model and understand the anti-cancer efficiency of your viral nanoparticles, which is loaded with the anti-cancer drug. So basically, we are converting the villain virus into the hero virus, that is the viral nanoparticle, so which can be specifically targeted to the cancer. So we got the overall idea about what is a nanoparticle and how to make the nanoparticle by physical method, chemical method, and biological method. So now I'll be talking about the cancer nanotechnology. So what is the major difference between the cancer and tumor? The tumor is localized. So it is not capable of metastasis. It does not kill the host. So by doing the surgery, we can remove the tumor. Whereas the cancer, it spreads from one location to other location. So the spreading process is called as metastasis. So and that will kill the host. So it will spread and it will kill the person. That spreading process called as metastasis. So the tumor can be removed by surgery, but the cancer is very difficult to cure. So that's what we need the early diagnosis. So if you see that the types of cancer, the 90% of cancer is mainly due to the on the carcinoma, that is epithelial tissue like glands and breast and skin. And if you see that the reason for this cancer, it may be due to the mutations, or it can be due to the environmental factors like UV or exposure to different chemicals or some kind of viral infections that will also convert your normal cells into cancer cell. So basically, what is happening inside the cell? So when this normal cell became a cancer cell, so each cell will have a proto-oncogenes, that is the potential for become a cancer cell, and it also has these tumor suppressor genes that will suppress the tumor. So this is like a kind of good character and bad character in all the person. So when you keep on torturing the good person, then he will show his okay bad character. So these two should be properly balanced, like growth promoting proto-oncogenes and growth restricting tumor suppressor genes. So this will be in all the cells. It will be properly balanced. If there is any imbalance, so that lead to the uncontrolled cell growth, that is a cancer. That is conversion of proto-oncogenes to oncogene, or there is a low expression of tumor suppressor genes. So this leads to the cancer. And the recent research shows that at least every day one or two cells are trying to become a cancer cell. Our immune system is going and destroying it. So if you're having a strong immune system, then that will also save you. You can see this is a cancer cell and this is the immune cell, and it will go and attack and destroy the tumor cells. So we can use these nanomaterials for cancer diagnosis. For example, we can use the fluorescent nanoparticles like quantum dots. So the advantage is with respect to the size, it gives a different kind of fluorescence. And again, the quantum dots, the major challenge is it is made up of heavy metals, and also it has a toxicity and solubility issues. So to improve the fluorescence, we can have the inorganic cell, which will improve the brightness and stability. And we can also have organic coating so that it provides a water solubility. And we can also add antibody so that it can specifically go and bind to the cancer cells. So you can see the difference between the quantum dot based diagnosis and organic dye, the traditional dye. So when you use the traditional dye, so when there is a high level expression of the particular cancer marker, so in the advanced stage of cancer, the traditional dye is able to give some signals. Whereas in the low level of expression, that is the early stage of cancer, so you can see that there is no signal. So when you use the traditional method, so the doctor may say that you are safe, there is no cancer. But when you use the your nanomaterial like quantum dots, even there is a low level of expression of the particular marker, that is early stage of cancer also, it can detect and it can give the signal. And as I told you, there is no crosstalk of fluorescence. When you use the traditional organic dye, maximum you'll have a red green fluorescence or orange fluorescence or yellow fluorescence, and there'll be always a overlapping of fluorescence. But here, with respect to the size, it gives a different fluorescence, so there is no crosstalk or overlapping of fluorescence. And we can also do the multiplexing. We can combine this cancer. For example, the cancers will express different kind of markers, right? So we can also do the multiplexing, and by using this, we can also do the early diagnosis. For example, we can have a nanoparticle tagged with the different markers, antibodies. So this is for red color fluorescence, this is for green color, and this is for yellow or green color. So, so what happened is like when there is a expression of this kind of marker. So if you see that if it is only the yellow color or green color fluorescence, we can say that is in the early stage of cancer. And if the all the fluorescence are expressing that, we can say he is in the advanced stage of cancer. We can see this in the mild stage of cancer, only the blue color is expressing. So in this case of advanced stage of cancer, all the colors are expressing. So it is binding to the all the markers. So we can also do the multiplex diagnosis for early diagnosis of cancer, because the cancer is not due to the one specific. It will show at least 2 to 3 different types of markers signal. So we can diagnose it and we can predict the disease. And this quantum dots was very stable. So that is the advantage of this using this quantum dots for the cancer diagnosis. So you can see here, when you inject into the mouse model with the tumor, it is specifically going and binding to the tumor, and only the tumor is glowing. And additionally, we can also use another method called chemical nose method. For example, you purchased a bag of apple, and if any one or two apple gone bad, okay, you can easily smell and tell which apple is gone bad instead of sending to the lab. Similarly, we can also make the polymeric nanoparticle that is called the chemical nose. So these nanoparticles are wrapped within a polymer, and this polymer is specific for the particular cancer cell receptor. So this is a cancer cell, and the cancer cell has the different kind of receptors. And for example, so this is for blue color fluorescence, and this is for green, and this is for red fluorescence. So if it comes and bind to the particular receptors, the polymer will be degraded and the fluorescence will be on. So when it is nanoparticles along with the polymer, the fluorescence is off. When it's bind to the cancer cell, then the polymer is degraded and the fluorescence is on. So this way, we can say that depends on the fluorescence on, we can easily diagnose the cancer. If all the green, red, and blue fluorescence is on, so that means he's in the advanced stage of cancer. If it is only one color fluorescence is on, we can say that he's in the early stage of cancer. So this is a kind of chemical nose. And again, in the cancer, the cells are divided rapidly, so there will be always some change in the temperature. So the temperature can be also measured using this nanothermometer. For example, whenever we go for jogging or walking, our body temperature rises. Similarly, in the cancer cell, divides faster, so there will be always the change in the temperature. So how we can detect it? We can also have a nanoparticle, and nanoparticle with the polymer, and polymer on the top of that you have the gold nanoparticle. So with respect to the temperature change, the polymer will expand, and it gives a different kind of the fluorescence or the color. So that can be measured. So that can be a kind of nano thermometer. And again, the simple the pregnancy test kit. Most of you know, it is based on this particular hormone overexpressed in the pregnant woman. That is a HCG, human gonadotropin. And if the particular female is getting 2 lines, that means she is pregnant. But the same pregnancy kit can also be useful for male. So if male is getting 2 lines, doesn't mean that he's pregnant, but he has a high chance for the testicular cancer. So during the testicular cancer, there is a high chance this will also overexpress in the male. So by just simply putting 1 drop of urine, so within 30-40 rupees, you can also diagnose the particular male is having the testicular cancer or not. So that is the advantage. So in our lab also, we are trying to develop such kind of paper-based, lateral flow assay-based diagnostics for bladder cancer and other cancers. So that is still work is going on. I'll tell you later. So we got the idea about how we can use nanoparticles for cancer diagnosis. So now we'll learn about how we can use these nanoparticles for cancer therapy. So in the traditional treatment, whenever you give the anti-cancer drug, so this the drug don't know which is normal cell, which is cancer cell. So this is like a kind of bullets or bomb. It don't know who is a good person, who is a bad person. So it will go and attack all the cancer cells as well as the non-cancerous cells. But in the case of nanotechnology, this nanoparticle is specifically go and bind and kill only the cancer cells, and the healthy cells, the normal cells are intact, so it is not affected. So that is the advantage. So it specifically go and bind and attack the cancer cells. And in the therapy, when you target this nanoparticle, there are 2 types of targeting. One is passive targeting, the other one is active targeting. So the passive targeting, whenever you make any nanoparticle of size between 70 to 200 nanometer, it automatically go and bind to the tumor location. And that is similar to that. Suppose you want to send a letter to your President of India or Prime Minister of India, you can simply put President of India, New Delhi. The letter automatically goes to the particular person. So that is called like a kind of simple example for understanding the passive targeting. And active targeting is you are sending the letter to your close friend, even though he's your close friend. Still, you have to write the complete details like the particular apartment number and name and including the mobile number and the pin code or zip code. Everything should be mentioned. So the active targeting, you'll be giving a specific antibody or specific peptide which can go on bind to the cancer cells. So how the passive targeting works? Where the tumor cells need more amount of blood and nutrients for its growth. So what will happen is whenever it makes the blood vessels rapidly, it leads to the lot of damaged or leaky blood vessels. So these leaky blood vessels, okay, so that leads to the this one. All the nanoparticle can be accumulated in the particular tumor location. So that effect is called as EPR effect. Okay, and all the nanoparticles will be trapped in the particular tumor region, and that will destroy the tumor. You can see here there is a lot of leaky blood vessels. These nanoparticles are trapped in this green color tumor region, and this is a healthy blood vessels. You can see that there is no gap, so nanoparticle is not entering here. So only it's entering into the tumor location and destroying the tumor. So this is called passive targeting. So we are not doing anything. We are making nanoparticle, polymeric nanoparticle between size 700 to 200 or 250 nanometer. So that will be automatically trapped in the tumor location. In case of active targeting, we'll be specifically adding the antibody. So then it will go and bind to the cancer cell and destroy the cancer cells. So we got the overall idea about what is nanoparticles and how to make nanoparticle by different methods. And also we understood like how we can use the nanomaterials for cancer diagnosis and cancer therapy. So the next few slides, I'll be talking about what kind of research work we are doing in our lab. So our lab is Nanobiotechnology Lab Research Group at IIT Roorkee. So we are making nanoparticles for various biomedical applications. So I'll start with the first work which we started whenever we started our lab. So this is the first paper we published from our lab. So that time we don't have much lab facility. So what we had is only the microwave oven for heating the coffee or tea. So by simply using the microwave oven, so we made the fluorescent carbon dot. Because the quantum dots, I told you, it is made up of heavy metals. So there are some drawbacks. So to overcome that, we made the carbon dots. So the carbon dots also, it's a zero-dimensional material. And it is any carbon source. Any carbon source, you burn it in a controlled environment, you can make the fluorescent carbon dots. So when you compare to the quantum dots, which is toxic, these carbon dots are biocompatible and very simple to make and highly water soluble and easy to add functional group. So that is the advantage of these carbon dots. And we used chitosan as a carbon source and polyethylene glycol 4000 as a stabilizing agent. And we use sulfuric acid as a dehydrating agent. And we just put in the microwave oven and heat it. And we got multicolor carbon dots. And this is a protocol optimized in our lab. You can also follow the same protocol and use the normal domestic microwave oven, and you can heat it for 20 seconds and 10 seconds off. So like that you have to do it until it become a completely dark brown color. Then you can centrifuge, then filter it, then you can get the completely, you can get the fluorescent carbon dots. And that time we don't have the cell lab, so what we did is we used the bacteria and we added on the bacteria. We can see that with respect to the excitation, it gives a different multicolor fluorescent nanoparticles. And in the next work, what we did is we, as I told you in the one of the slide, the charge of your nanoparticle is very important. If we have a positive charge nanoparticle, so it can easily bind to the negative charge cells. So what we did is we use the chitosan and PEG, so which gives the negative charge nanoparticle, and we used PEI, so it gives a positive charge nanoparticle. So again, we made a simple experiment for understanding the charge. What we did is we made a gel, and we made the well in the middle of the gel. So you can see the well is here. So we loaded this and connected to the power supply, and all the positive charge moved towards the negative charge, and all the negative charge nanoparticle moved towards the positive charge. So, and it's a fluorescent carbon dots without any ethylene bromide staining or any other staining. We can also see the fluorescent carbon dot. And we also use the cell lines And by the time we got the cell lab ready, so we can see that the PI, which has a very good fluorescence, and when compared to the PEG, which is a negative charge. So negative charge nanoparticle binding is less, and positive charge nanoparticle binding is more. So we combine these therapy and diagnostics, and we made a theranostic nanoparticle. So as I told you earlier, therapy plus diagnostics, that is a theranostic nanoparticles. So what we did is we taken this carbon dart. So the carbon dart is negative charge, and the silver nanoparticle is a positive charge. So we combined and made a kind of nano composite, and when you add it to the cells, cancer cells, it is inducing the cell death, apoptosis. And also additionally it has a fluorescent carbon darts. We can understand the cellular uptake of your nanoparticles. That is the advantage. So you can see here, when you use the only carbon darts, the cells are green color, the cells are intact. Okay, the cells are intact. And when the cells undergo apoptosis, it become rounded. So only carbon darts cells are intact. When you use the carbon darts plus metal nanoparticle composite, you can see with respect to the concentration, the cells are undergoing cell death. So that's why the cells, from the lengthy cells, it become like a rounded dead cells. And when you see under the electron microscope, you can see the breast cancer cell and lung cancer cell, untreated one and versus treated one. So the treated one is showing the apoptotic cell membrane blibbing. And most of the metal nanoparticles induce cell death by producing the reactive oxygen species. And the reactive oxygen species can be measured by using a simple chemical dye, that is a DCFHDA, which is a non-fluorescent. But when you add the this one metal nanoparticle to the cell, so that will produce more amount of reactive oxygen species, and that will convert your non-fluorescence into fluorescent nanoparticle. So inside the cell, there is a cellular esterase, so which will convert your DCFDA into DCFH, and this ROS convert this non-fluorescence into fluorescent. If we have more fluorescent in the cells, that means more amount of reactive oxygen species is produced inside the cell. So that can be studied by microscopy, or it can be studied by using the flow cytometer. You can see here in the control cells, the fluorescence is very, very less, the background fluorescence. Whereas when you treat with the different concentration of nanoparticle, you can see that the higher concentration, you can see that the amount of fluorescence is increased. So the next work is like we use the polymer for the delivery of the anti-cancer. For that, we use the dendrimer. So dendrimer is a kind of it's a Greek word that means dendron means it's like a tree-like structure. So the dendrimer will have like a tree-like structure. So in this, what we did is we added the anti-cancer drug called epirubicin. So epirubicin have a red color fluorescence, and on the top of that we added the carbon dart, which is a green color fluorescence. So inside we have a red color fluorescence epirubicin drug. Outside we have the fluorescence, the green fluorescence carbon dart. So this kind of a theranostic nanoparticle you can see here, and when you use the dendrimer, so it escapes from the lysosomal degradation. So the nanoparticle is taken inside the cells, and through endosome, the endosome will fuse with the lysosome, and in the lysosome we'll have a lot of positive charge. So due to that, the nanoparticle will burst the lysosome and release intact. So this effect is called as proton sponge effect. And we can also see that once the nanoparticle is attached to the cell, it will give a green color, and once the drug is released, it gives a red color, because this drug is specific for the nucleus. So once the nanoparticle attached to the cell, it gives the green color. Once the nanoparticle is attached and release the particular anti-cancer drug, and it binds to the nucleus, so you can see the nucleus will appear red color. So this nucleus will be red color, and the cytoplasm will be green color. So we can easily understand the nanoparticle entry and drug release, everything, by using this simple nanomaterial. So this is also highlighted in the article. And followed by that, what we did is we use a natural nanomaterial, that is the protein-based nanoparticles. So the protein-based nanoparticle, we use albumin, that is the BSA, bovine serum albumin, or we can use the human serum albumin. So the advantage of using the protein nanoparticle is it has a hydrophobic and hydrophilic packets, and most of your anti-cancer drugs are hydrophobic in nature. Okay, so if you can see that, like the FDA approved the first albumin-based nanoparticles, that is Abraxane. And before that, people are using this Taxol. So to dissolve 6 milligram of paclitaxel, we have to use this much amount of organic solvent. Whereas in case of Abraxane, you can see that we can use 900 mg of albumin and 100 mg of paclitaxel. So this is only 6 mg. Whereas here it is 100 mg of anti-cancer drug, and we are using only the protein, and we are not adding any surfactant or solvent. So you can make protein nanoparticle loaded with the anti-cancer drug. So additionally, the advantage is the cancer cells need more amount of protein for its growth. So this is like mixing the food with the poison. So when your cancer cells need a more amount of protein for its growth, and you are having a nanoparticle which is loaded with the anti-cancer drug, so it will take up more amount of protein nanoparticle, and that leads to the death of the particular cancer cells. So we use the BSA and we use the niclosamide. Niclosamide is a basically it's not a exactly anti-cancer drug. It is a drug for killing the tapeworm. So that's anti-helminthing drug, and we did a drug repurposing. So we use a niclosamide, and this is a hydrophobic drug, and which is packed inside the BSA nanoparticle, and it also have a very good anti-cancer effect. So you can see here, usually the drug is like a rod shape, and when you make it into protein nanoparticle, it becomes spherical. The size is between 200 nanometers, so it can be taken up by the passive targeting. That is the EPR effect. That is advantage. And you can see that when we add the free drug is not toxic. And when you put it into the nanoparticle, so you can see that it is taken up by the cells. The cells is undergoing cell death. And we also calculated the IC50. IC50 means the concentration required to inhibit 50% of the growth of the cell. So you can see here, so this is a 40% and this is a 50%. So roughly 5 micromolar is required for killing the lung cancer cell lines. The same nanoparticle for killing the breast cancer cell lines, the IC50 concentration is different. So you can see here the IC50 concentration is roughly around 2.5 to 2.6 micromolar. And the other work, what we did is we used the nanofibers for loading the anti-cancer drug. So to give a brief idea about what is nanofiber is a continuous fiber, and which has a diameter in the range of nanometer. And you can see that to give us rough scale difference, this brown color is a human hair. So this is a human hair. Okay, so this is a human hair, and the below this is a nanofiber. So how much small is the nanofiber? You can compare with this brown color human hair. So how to make this nanofiber? There are three approaches. Out of that, the electrospinning is one of the simplest approach for making the nanofiber. So the principle of electrospinning is similar to the cotton candy. So here we'll be putting the sugar crystal, and it will melt and forms the fiber, basically using the collector and collecting the particular fibers. So the electrospinning principle is similar to that. We'll be adding the polymer instead of sugar, and instead of pink color, we'll be adding the anti-cancer drug. And when you apply the electric current, you can see the fibers will be collected in the collector. And this is how the final product look like. So this is the curcumin, that is a haldi turmeric loaded anti-cancer drug loaded nanofibers. That's why it's look like yellow color. So this can be useful for controlling the tumor reoccurrence after surgery. For example, whenever the doctor do the surgery and they remove the tumor, so if it is not completely removed, it can reoccur and create issue later. So to overcome that, they can use this kind of bio bandage. So once they remove the tumor, so they can put this bio bandage, which can act as a kind of physical control, as well as it also relieves the drug in a slow and sustained way and control the cell growth. So what we did is we added two different types of anti-cancer drug in the fiber. There is the core shell nanofibers. So here we added one type of anti-cancer drug, and in the shell we loaded another type of anti-cancer drug. So when you have two different types of anti-cancer drug, so for example, one will attack the nucleus, and the other one attack the cytoplasm. So different pathway. So it will have a synergistic effect, which will have a better anti-cancer effect. And you can see the core shell morphology under the this electron microscope. We can see that. So you can see the nanofibers and the core shell morphology under the scanning electron microscope. And whenever we make the nanofiber, we have to make sure this nanofiber membrane is hydrophilic in nature. So for that, we can use a simple experiment called contact angle measurement system. So you can take the nanofiber membrane, put a drop of water, and take a photograph, and using the software measure the contact angle. If the contact angle is less than 90, the material is hydrophilic. More than 90, hydrophobic. More than 150, it is super hydrophobic. A simple layman example is if you put a drop of water on the plastic sheet, the water will be intact, that is hydrophobic in nature. And if you put the water on the paper, it will absorb, so that is hydrophilic in nature. So we need the nanofiber membrane which should be hydrophilic in nature so that it can be attached to the body and it can also degrade inside the body. So you can see here, this is a only nanofiber, and after loading with the anti-cancer drug, as you know, this anti-cancer drug is hydrophobic in nature. So the slightly the contact angle is increased, but it is still within this less than 90. That means material is hydrophilic in nature. This was also highlighted in the Nature India. And the last work, what I'm going to show is like we made a theranostic carbon darts. Instead of making carbon darts separately and metal nanoparticles separately and combining it, so why don't we make bioactive carbon darts? So for that, we use this plant extract, plant leaf extract. That is, it has a lot of Vinca alkaloids, and it has a high affinity towards the tubulin. So we made a fluorescent carbon darts from this plant leaf extract, and it is highly specific for the microtubule. It will attach to the microtubule and it will depolymerize the microtubule and destroy the cell. So these microtubules are like a support for your tent. For example, you are making a tent and you are putting this support. So these microtubules are the support for your cells. If the support is removed, the cells will fall down. The tent will fall down. Similarly, the cells will die. So we use the hydrothermal method. That means 200 degrees Celsius for 10 hours. And this complete leaf extract will become like a dark brown carbon darts. And when you see under the UV light, you can see the fluorescence. And so when you see under the confocal microscope, this is not stained with any other stain. It is carbon darts. You can see here with respect to the excitation emission, you can see the different kind of fluorescence. And it is binding to this carbon darts are binding to the microtubule, and microtubule is a depolymerization happening. And you can see the disintegrated tubulin labeled with the carbon darts. So the microtubule depolymerization is happening and it is destroying the particular cells. And this work also highlighted in the Nature India. So at the end of my talk, I'll be briefly telling about the NPTEL course. I'm also teaching two NPTEL course, this biomedical nanotechnology, usually in the autumn semester. And this will be in the spring semester. But if you want to take this course, biomedical nanotechnology, this is also available in the spring as a self-study course. So anyone can take this course, students or faculty, because these are the AICT approved course. So we can take this course. So additional advantages, recently this NPTEL, they introduced the NPTEL pre-doc fellowship. For example, your students, masters or BTech students, and if they are qualifying this 3 NPTEL course, okay. So the 3 NPTEL course with the 60% mark, and also they are submitting all the assignments on proper deadline. So they will get an NPTEL discipline star, and they have the opportunity to do internship at IIT. It can be any IIT. It is not only to me. It can be, they can go to any IIT. So there'll be they have the choice to select 5 IITs, and there'll be a small interview or a discussion. Then the particular faculty will select the candidate. So there is no need for any gate exam or anything. So they have to qualify 3 NPTEL course with the 60% mark, and they'll get a monthly stipend of 25,000 plus 10% HRA and contingency grant of 1 lakh per year. So they can use it for going for conference or purchasing the chemicals. And after doing the project with a particular faculty, so they can also join a PhD if they are able to qualify GATE or any national level exams. So last year also one student came and joined with me. So during that time he don't have any GATE exam, but he was doing project with me. Then he qualified the GATE. Then he applied for the PhD interview and he got selected and he joined PhD with me. So this is another opportunity for students to get into PhD into IIT. And another student also joined with me, but he also got a good publication during this 1 year, and he is going to abroad for his PhD. So by both the way, the students get benefited. So you can make use of these fellowships, and you can also inform to your students or your colleagues, so they can also register for this course. So this NPTEL will give different kind of star badge. They'll give a discipline star, or if you're taking more than 3 or 4 courses, they give the different kind of certificates. So all these certificates, the star badges are eligible for going for this NPTEL internship. So with this slide, I'll conclude my talk. I thank you all for your patience and listening, and I'll be happy to discuss and answer. Okay, and our institute, IIT Roorkee, we have almost, it was established in 1847. So it's more than 178 years old institute. So, and this building is more than 178 years old building. So whenever you get chance to visit IIT Roorkee, you can visit this building as well as visit our lab. Okay. So thank you all. I'll be happy to discuss.
So thank you, sir, for your illuminating lectures. I think the lecture will explore us to diagnosis and current therapies with emerging field of nanotechnology. And now few questions can be addressed here. One fellow, Dr. Ajay Kumar Das, has put a question. So, may I read? Maximum nanoparticles are toxic in nature for living systems. How much it is advantageous in terms of its cytotoxicity?
Okay. So the cytotoxicity will study before we take it to next level. So that's what we are using the polymeric nanoparticles. So polymeric, again, we use the FDA approved polymer and FDA approved anti-cancer drug. So in this way, we can avoid the toxicity, and any drug will have a toxic effect. We have to select the right concentration, which has more therapeutic effect and less toxic effect.
Okay.
Yes.
Dr. Rajesh Ghosh has raised his hand. You can ask question. Dr. Rajesh Ghosh. Okay. Okay, another fellow.
Sorry. Can you hear me?
Okay,
Yes, yes.
okay.
Okay. So
Yes,
thank you,
sir.
sir, for your very exciting lecture. So one question, you were talking so much about the carbon dots.
Mhm.
So the same questions like the ma'am, you were using carbon dots for so many times, and uh, it will generate lots of reactant oxygen species ROS. So if it is more produced, then is there any effect on the healthier cells? Because we know that the lots of ROS may harm the normal healthier cells along with the cancer cells. What about your opinion, sir?
Yeah, basically the carbon dot is biocompatible. We'll combine the carbon dots with some metal nanoparticle, and we'll be using the optimum concentration which can kill. And again, we'll be targeting these carbon dots specifically for the cancer cell. So this way, we can reduce the toxic effect to the healthy cells. When
Okay.
compared to the traditional medicine, where all the cells get attacked by the anti-cancer drug, we can reduce the toxicity and we can improve the therapy. That is the advantage of this nanoparticle.
So you mean that it is a
Yes.
very targeted delivery to the cancer
Correct.
cells only?
Correct. Correct.
Okay. Okay. Thank you. Thank you.
Yes.
Okay. Thank you.
So another fellow, Dr. Orijit Das, has question: is there any negative effect of using viral coat as drug delivery system? Also, is there is a possibility of reactions between viral coat and the drug inside?
Okay, so that's what we are avoiding the animal virus. We are using the plant virus. So there is very, very less chance for inducing the immune response. Otherwise, if it is the particular nanoparticle is coated with the polyethylene glycol and these things and targeted, so that can be degraded. And again, the plant virus, all these viruses are highly specific for the particular. So that's why we are using the plant virus to minimize the immune response. So this way we can overcome that. And while nanoparticle again, so it will be targeted and it has a lot different kind of shape. So it will also have a more therapeutic effect.
Okay, thank you, sir. Now, Dr. Ornisha Chaudhary has raised her hands. Anisha Chaudhary, please.
Yes, ma'am. Thank you, sir, for your very nice presentation. Very interesting topic. Sir, I have one query. That is, we have learned from here that quantum dots are developing technology which uses nanoparticles for highly sensitive multicolor imaging to detect the cancer cells. PET scans are routinely used, which uses radioactive tracer elements to locate metabolically active cells, which enables us to detect the cancer cells. Then why these quantum dots are not routinely used for therapy for detection purpose? Can you please let me know?
Yeah, the quantum dots still in the R&D. Anything to take it to human trial, the clinical trial will take more time. So they have completed in the animal studies. So that's what like they will be taking to the next level. But they found that the quantum dots are toxic. So that's what they are now moving towards the carbon dots and other fluorescent material. So it is still in the animal studies and the clinical phase 1, 2. So it will take some more time to come into the human application.
Okay, okay, sir. Okay, okay. Thank you so much.
Okay.
Okay.
Okay, thank you. Now, Dr. Normata Roy perhaps has some question. Dr. Normata Roy, please. Okay.
Thank you, ma'am.
She has put
Sir,
question
thanks a
in.
lot. Okay, I'll directly ask, sir. Sir, thank
Yeah.
you for your very engaging and lucid lecture. And I must say the examples that you used were really interesting, especially the one about, I like the one about the passive and active therapy, about writing a letter to the president, and one to your close friend. Uh, what
Okay.
I wanted to ask is that in the chemical nose method that you
Mhm.
mentioned, that nanoparticles are coated in polymers, and when the
Mhm.
polymers go bind to the cancer cells, the polymers are dissolved or they are removed. So my
Right.
question is, are the polymers designed in such a manner that they are receptor specific, so that they go and particularly bind to the cancer cell?
Yes.
And
Yes.
after binding, how are they removed? I mean, how is the polymer dissolved away and the nanoparticle comes in contact with the cell?
Yeah, basically it will bind to the receptor. It will degrade by the normal enzymatic. So, or pH based. For example, the tumor location has the acidic pH. So, it is a kind of smart delivery. So, in the acidic pH, that will be removed, and it will bind and it will release the anti-cancer drug. It can be for smart delivery, or it will bind to the particular receptors. Then automatically the polymer will be ruptured. So then it can give the fluorescent on. That way it is designed.
Thank you. Okay, thank you, sir.
And there's one more question like positive charge, negative charge, and zeta potential. That means the if it is a positive charge, the nanoparticle is positive in nature. So if you the more positive charge means the nanoparticle is positive charge. You're getting zeta potential negative charge. That means it is nanoparticle is negative charge. And one more question on
Okay.
nanoparticle entry into the cell, like it will attach to the cell receptor. For example, in case of breast cancer, so the breast cancer will have the folic acid receptor. So we can add the folic acid to the nanoparticle. So the folic acid will go and bind to the folic acid receptor on the breast cancer cell, and it will release the anti-cancer drug. It will be taken by the endosome. It goes to the lysosome. It forms the endolysosome. Then it will degrade and release the anti-cancer drug, and it destroy the cancer cell.
Okay. Okay. Durba Bhattacharya has a question. Could you elaborate on the entry of nanoparticles into the cells, and what are the conditions which will ensure entry of nanoparticles at targeted cells?
Please could you repeat it? What is it?
Could you elaborate on the entry of nanoparticles into the cells?
Yeah, yeah, that's what
And
I
secondly, what are
previously
the
I told.
okay, okay, okay. Yes, yes. And Abhijit wishes, how can hydrogels derived from polymers or peptides can be classified as nanomaterials? I think is a elaborate one.
Okay, so the hydrogel is not a nanomaterial. We have to use the nanogel. If the hydrogel in the nano size, then
Okay.
it can be called as nanogel. So as I told you, any one of its dimensions should be in the range of nanometer. Out of this X, Y, Z, okay, any one should be in the range of nanometers. So then we can call it as a nanoparticle.
exactly. Dr. Debulina Sharkar has raised her hands. She has some question. Dr. Debulina Sharkar.
Yes, ma'am.
Yes.
Thank you, sir, for your lecture. I have one question. I work in metal ion sensing field. So, if I incubate the cells with metal ions, they penetrate inside it, and in presence of certain chemosensor, it is showing bright fluorescence.
Hmm.
But my question is that can you give me an idea that whether is it possible to quantify the amount of metal ion inside the cellular region?
Yeah, if it is tagged with some fluorescence, you are telling some it is giving some fluorescence, right?
Yes, but how much of the metal ion has actually crossed or penetrated inside the cells?
Okay, okay. So what you can do is you can gently wash the cells, and the whatever attached in the surface will be removed. Then you take the cell and lyse the cell, and you can do the ICP-MS. So whatever the inside metal that can be measured, quantified.
Okay, okay, okay. Okay, sir. Thank you. Thank you, sir.
Okay. Okay.
So Dr. Abhishek Basu has question. He asked, for testicular cancer detection kit, what kind of molecule is supposed to be detected in the urine?
Okay.
And is there any specific kit being developed for the easy detection of prostate cancer?
Okay, the same HCG which is in the pregnant woman urine. So that is same HCG in the testicular cancer patient. So nothing to do. You can purchase a normal pregnancy test kit and put the urine sample and check it. But for the prostate cancer, we have to use
Okay.
the prostate specific antigen, the PSA antigen. So that kind of kit is
Exactly.
yeah, it is already available, but it's not like a point of care test. It has to be go to the hospital and you have to do it. But in our lab, we are trying to make
Exactly.
it like same like a point of care test, like a simple urine
Oh,
based. But still, it is in R&D, so hopefully we'll be able to do.
exactly. So, okay. Sir, exactly so that human chorionic growth hormone, the
Yeah,
gene responsible for producing that, if it is overexpressed in male, that can be a indication of prostate cancer or testicular cancer.
it is for the testicular cancer.
Yes, yes.
So the prostate cancer,
And
there are
for
some other.
prostate, the indication is PSA
Yes,
level,
yes. Yeah, prostate specific antigen.
prostate specific protein or antigen. Exactly.
Yeah, there's one more question
Okay.
on how
I
RGD
think
peptides are attached. So RGD peptides
exactly.
can be
Mhm.
attached by simple functional functionalization. Like we have a nanoparticle. Suppose if we have a protein nanoparticle, which will have an amino group, and we can add the carboxyl group of this. So carboxyl group and amino group, it can be connected by this chemistry called EDC NHS chemistry.
Okay. Dr. Manushi Mondal has a question. Is you have using filamentous green algae for producing nanoparticle or nanomolecules? Can we use that?
I'm not using, but there are some research group they're using the green algae, and yeah, you can also use the fungi. You can use any kind of even you can use the curd.
But
Curd also has a lactobacillus. You can use that for making the
okay.
nanoparticle.
I
But yeah,
think plant virus is more useful than algae or other
yeah,
like materials.
algae we can make the
Yes,
nanoparticles.
yes.
Bio since the metal nanoparticle can be done. Virus we can load the anti-cancer drug.
Okay. Okay. So I think I have covered all the questions.
Yes. Someone has the
If there is no question, then we must
Arunima.
sum up here.
Yeah,
So thank you, sir.
madam, I
Yes.
have a question to ask, sir.
Okay.
Thank you. Sir, it was a very
Mhm.
uh, illuminating lecture, I must say. But one thing I want to ask everybody who is working on nanoparticles or nanoparticle aided
You are
drug
not audible.
about, hello,
Yeah, yeah, audible. Yeah, yeah.
Please.
that they're
Yeah,
talking
yeah,
about
yeah.
cancer therapy itself. But what I wanted to know is there are certain conditions which are related to reproductive ailments.
So can we design a drug that can be used as an oral contraceptive, and we can use nanoparticles to deliver to the specific gonadal regions? Because I have worked on certain phytoestrogenic chemicals which hinder hinder the reproductive pathway in a different kind of a way, in a dose dependent manner. It can act as it can act as a for positive feedback as well as negative feedback control of certain endogenous hormones, specifically estrogens. So, is it possible to design a nanoparticle, kind of taking the viral coat from a plant extract? Because it is a phytoestrogen, there should be minimal kind of crosstalk between the two
It's
chemicals. So is
possible,
it
ma'am. That is possible. We can also use the polymeric nanoparticle also. We can also use the hydrogel or nanogel also, and which can be released with respect to the like a pH, or it can be released. That is possible to make. Target delivery is possible.
thank you, sir.
Thank you. So there's one more question, like how do biopolymers like albumin differ in their advantage? Yeah, so
Exactly. Compared to
albumin
synthetic
highly
dendrimers.
compatible, highly biocompatible material, albumin. So there is no side effects. So that is the advantage. And again, the polymer also, it is also biodegradable polymer. So both has the own merits and demerits. So the advantage with the dendrimer is it is a positive charge polymer. So it can the binding efficiency is more, and also the delivery of drug is more. And again, the protein nanoparticle, the advantage is like the cells will assume that protein is a kind of nutrient, but the particular nutrient is mixed with the anti-cancer drug, that is a poison. So there is a both the way it has a both has its own merit and advantage for killing the cancer cell.
Yes. So, uh, Narmata Roy has another question. For protein entries, does it have to be specific protein for a specific cancer cell? Or do they take all kinds of proteins non-specifically?
Proteins, so far, yeah, we can use this basically the human serum albumin for human application and BSA. So these are well established methodology, and already in the clinical also it is approved. So that's what like we can use this protein based nanoparticle, and the protocols are very optimized. For other proteins, so far some very less number of work is going on. So the protein BSA will be more easy to translate into the next level.
Okay, so thank you, sir, very much for your illuminating lectures and addressing all the questions. I think all the participants are very happy, and we get much benefit out of your lectures. And this is a very current topic and current area of research also. And we are running very short of time. Our next speaker is waiting. So thank you,
Okay.
sir, for
Thank you, ma'am. Thank you very much.
spending your valuable time to be us. We will again contact you whenever we required in this session.
Surely.
Okay,
Thank you.
sir.
Thank you very much
Sir.
for the wonderful opportunity. And also, I thank all the participants for the engaging discussion. So, very active participants. Thank you
Exactly,
very much.
sir.
Thank you all.
Thank you. Thank you, sir. Thank you so much. Thank you so much. So we are now proceed to our next lecture. The next lecture will be delivered Dr. Shubhit Chandra Dasgupta, retired Professor of Zoology, Honorary Emeritus Professor, Department of Zoology, Ramakrishna Mission Vidya Mandir, Belur Math, Howrah. He will deliver lecture on nanotechnological assessment of venom-induced nanotoxicology. Dr. Dasgupta is presently associated as Honorary Emeritus Professor at Ramakrishna Mission Vidya Mandir. He was served our postgraduate Department of Zoology, Maulana Azad College, over a decade. He has obtained his master's degree in zoology in 1981 from Calcutta University and PhD in 1989 from School of Tropical Medicine. Beginning his career as lecturer in Darjeeling Government College in 1985, by dint of dedicated work and his passion for research, he steadily rose to become professor in 2010. During his tenure of work, he has published more than 50 research articles in peer-reviewed international and national journals, 3 books, 2 book chapters from international publishers like Springer and Taylor Francis in his area of teaching, and more than 45 conference presentation and 1 international and 1 national patents. This year, Dr. Dasgupta got an assignment from famous publisher Elsevier and acting as an editor for the book Snake Venom and Human Health, translating nature's toxin into therapeutics. He has successfully undertaken many research projects sponsored by national bodies like UGC, DST, DRTO, CSIR, NTRF. Professor Dasgupta is active member of many scientific bodies which includes Indian Science Congress, Indian Pharmacological Society, Indian Biomedical Research Association and Zoological Society of Kolkata, and task force members of DBT Star College Scheme, Government of India. Being a government college professor, he is the nominated government member and representative in many academic bodies in different universities. Till date, he has guided 13 PhD students. Dr. Dasgupta research mainly includes the bioactive compounds from natural resources and its biological characterization with therapeutic assessment. Dr. Dasgupta, please. Sir.
Yes, thank you very much. Before start, I must express my sincere thanks and gratitude to Principal Maulana Azad College and all my colleagues, Coordinator Professor Deepak Kumar Shom, the introducer of Dr. Shormishta Bonik. All are known to me very much because I retired from Maulana Azad College in 2023. So once again, I convey my gratitude to all the persons who consider my name as a resource person for this online refresher course. As the online refresher course, its title is oriented along with the nanoscience, and being basically a zoologist, so my introduction in the field of nanoscience with the help of the venoms, right? I basically worked with the venoms and the toxins and the animal byproduct since for the last 25 years. So with the snake venom, I have some experience of the nanoparticle tagged venom and to reduce their toxicity. So that is my exposure in the field of the nanoscience. So I have, as I have authority assigned me 2 lectures, so I broadly differentiate my lectures. One is what is nanotoxicology, what are the properties of the toxic particles, say for the gold particle, nanoparticle, lead nanoparticle, silver nanoparticle. So what are the properties of that particle that causes the toxicity of a living being? Here we worked in the living model, the mice and the rat. So how we can assess the nanoparticle causes the toxicity of the living organism. So this is the first part, property. And what are the avenues nowadays and the recent avenues regarding the application of the nanotoxicology in the field of the venom research. As we all know that conventional venom therapy is an anti-serum, that is venom injected in the horse, and after 1 month a regular dose of injection of venom. The antiserum collected from the horse, and from there this immunoglobulin has been separated, and that is till now in India that conventional therapy continues. But with the advent of this nanoscience, this concept has been totally changed, and there are new methods have developed regarding the nanoscience, application of the nanoscience in the venom research also. So the broadly two parts of my lecture, one is regarding the nanotoxicology, nanoparticle toxicology, and the second part, what I have done in my research career with the nanoparticles along with the snake venom. So this is the broadly two parts. Today we are going to discuss about the only the toxicity of the nanoparticle, and the second day, that is on 10th, I'll discuss my work with the nanotoxicology and the venom research, right? So I just want to share my slide, just a minute. Slides are visible?
Yes, sir.
Yes, sir. Yes.
Okay. So the nanotechnology, the first thing, the line is small is beautiful, danger comes into small particle. So the nano means it is a very small particle, but there are a lot of advantage of these use of this technology, but this nanoparticle often causes a serious effect inside the living body. So the line is more appropriate. The small is beautiful, dangers comes into the small particles. So a quick recall of the history. This 1959 on December 29th, Feynman is the first person who told about the nanotechnology at an American physical society. But before 1959, actually the nano particle concept, we usually, the Indians are familiar about the homeopathic treatment, right? All of us know a little bit about the homeopathic treatment and the different dose, 1x, 2x, 6x, 30, 60, 200, like this. So what is the actual reason of this making this so many doses? So that is the original of the concept of this nanoparticle. Later on, after this concept developed through by this Feynman in 1959, this area gains much attention, and naturally now till date there are tremendous development of this in the field in different aspects. So the scientist or the professor Norio Taniguchi of the Tokyo Science University introduced the term nanotechnology. Now come to the nanoscale. Nanoscience deals with the research and technology development at 1 nanometer to 100 nanometer ranges. So nano means indicate 10 to the power simply minus 9. And nanometer means it is 10 to the power 9 nanometer. So a single human hair, if you compare with the human hair, is around 8,000 nanometer. So a typically protein size lies between 3 to 10 nanometer, and the red blood cell, which is a very model cell for every experimental analysis, the standard size is about 6,000 to 9,000 nanometers. So I have given this many data to compare what are the tiny particles ranges, that is nano, that indicates 10 to the power minus 9. So I have given the title of the slide, the nanoscale. Now come to the toxicology, because toxicology is a broad area, and those person who are exclusively deals with the animal poison and the venom, as myself, the area nowadays has been more classified as a toxinology, not toxicology, because toxicology, that means arsenic poisoning, lead poisoning, etc., etc. Heavy metal poisoning is a toxicology, right? And but if it is done with this carried out with the animal poison and the venom, then it area comes into the toxinology, a new area of the biological sciences. But as a whole, That includes under the nanotoxicology. So it is a branch of bio-nanosciences which deals with the study and the application of the toxicity of the nanomaterials. So preliminary, we are more interested to know about, after introduction of the nanomaterial inside in a living body, what are the condition of that nano particles. That means in vivo toxicity, how we can determine the in vivo toxicity. In our experimental analysis, the simple method to judge the toxicity is that to carry out LD50 or MLD, right? So LD50 and MLD is a standard method by which one can ascertain the what are the doses of the toxicity. But in case of the nanoparticle, the method is absolutely different, and itself that causes severe alteration in the human body. But interestingly, at the very beginning, I just told that the nanoparticle conjugated venom, that plays a very important role to reduce the toxicity of the venom. So in that point, it is somehow ambiguous, and I will try to explain why this ambiguous property of the nanoparticles arises. Specifically for the venom tagging, we use the gold particle, silver particle, but I am not in a position to analyze all the nanoparticles at a glance. I do not know if it is tagged with a titanium, right? What are the different aspects of the toxicity? That is a separate issue, but we'll try to show you what are the fate of the gold nanoparticle, silver nanoparticle in terms of the toxicity. So nanotoxicological studies are intended to determine at what extent their properties may pose a threat. That is an important line. So at what extent their properties may pose a threat to the environmental and the human being. Initially, this toxicity can be analyzed in two aspects. One is environmental aspects, another is from the biological aspects, right? So the particle also released in the environment either through water or through the soil or through the effluent from the different factories. So different aspects. One is causing the damage to the environment. So the nanotoxicology in relation to environment, and nanotoxicology in relation to the biological system. But the important is that, so intended to determine at what extent their properties may pose a threat, at which level that causes the threat to the organism for its considerations. So terminology, that means nanotechnology, nanoparticle, and nanomedicine. Nowadays, as all of you have a slight idea what is a nanomedicine, I will discuss a very short in brief the nanomedicine, because the Venom Research Group, recently Indian Institute of Science, Bangalore, a few scientists are trying to produce the nanomedicine, nanorobots, instead of the antiserum therapy, right? So this is a recent trend of the snake venom research. So conventional antiserum therapy and storage of the antiserum in the rural hospitals where there is no electricity, that pose a very problem. So efficacy of the antiserum arises a question. So with for that focus, so nanomedicine, nanorobots comes into the pictures. So if that toxicity can be reduced with that nanomedicine, is a very upcoming branch. So the technology, the use and the application of the material with the size in the nanometer range, particle, a microscopic particle of the matter that is measured on the nanoscale, and medicine, the branch of the medicine concerned with the use of the nanotechnology. So simply the advent of the medicine. The toxicity of the nanomaterial were broadly classified. I have already previously told the biological toxicity and the environmental toxicity. So I am concentrating about this biological toxicity, because nanotoxicology is an aspect of nanoscience that deals with the study of the adverse effect of the engineered nanomaterial or nanoparticle on the living system. Why it is engineered? Because in the laboratory we have to produce the nanoparticles. So in that case, I have used the term engineered nanoparticles. The ever increasing application of engineered nanoparticle for biomedical application has raised serious concern about their safety in human or any other organism, because as we do not have any human ethical clearance, so all the experimental study are carried out in the mice model, so in their safety in the living organism. And nanoparticles are widely used as a nanomedicine and nano carrier of drugs, what I have said, the nanorobots, due to their small size and exclusive properties. So they belongs, or they contain some exclusive properties. However, their size, morphology, surface functional group, dose dependent property may also be responsible for their toxicity towards normal, towards the healthy human cells or any living systems or any tissue or any organ. There are several studies have shown that chemically synthesized nanoparticles have high toxicity on live cells, that is biological systems, due to their presence of synthetic chemical. If you use, say, for the gold itself is a toxic. So if I carried out a gold nanoparticle, I have shown later on my slides how the gold particles can be tagged and with the venom, and that gold particle may too reduces the toxicity of the venom. But if you consider the gold toxicity, the chemically synthesized nanoparticle have a high toxicity on the living organism due to their presence of synthetic chemicals as surface functional capping agent compared to biosynthesized nanoparticle that possess biocompatible surface functional group. So if you synthesize the nanoparticle, biosynthesize the nanoparticle, that is more applicable rather than if you just an order there in our present system. If anybody would like to work with a nanoparticle, he may order the nanoparticle conjugated from Sigma. This is a famous chemical company. But that causes a severe toxicity. We have tried in our laboratory, but that particle is not very good to neutralize the venom toxicity. So what we have done, we inside the laboratory, we carried out and prepare a gold nanoparticle. With that nanoparticle, we have carried out several experiments, including the physiological experiment to evaluate the toxicity of the biosynthesized nanoparticle. So biosynthesized nanoparticle also exhibit a little toxicity upon reaction with the cells while disintegrating to its simpler form or due to accumulation. So the scope of the nanotoxicology is aimed to identify the potential hazards that are useful for the safety evaluation of the nanomedicine. So biological toxicity, the nanostructure can enter the body via 6 principal route. It may be intravenous, it may be dermal, it may be subcutaneous, inhalation, intraperitoneal, and oral. And interestingly, the nanostructure route is very much important because the toxicity may differ with the route. The entire nanostructure can distribute to various organs in the body. It may be goes to liver, lungs, brain, kidney, whatever may be the organ, and may remain the same structurally, be modified or metabolized. That means this line, significance of this line is that if you inject the gold nanoparticle tagged with the venom, if you inject the mice to assess its toxicity, so it initially goes to, if the venom is a cardiotoxic, if the venom is a neurotoxic or myotoxic, it initially goes to the brain or heart or kidney or liver or lungs. But the modification, structural modification of the nanoparticles may differ organ-wise. That line says clearly the entire nanostructure can distribute to various organs in the body and may remain the same structurally, be modified or metabolized, or it can be metabolized. So one can easily ask if we go for a pharmacokinetics study for these nanoparticles, what we have already known, the biotransformation process of a general drug, phase 1, phase 2 model, So the kinetic study can reveals what are the fate of these nanoparticles, right? Till now, we have not able to carry out that toxicity of the pharmacokinetics toxicity of the nanoparticle. But I think for other compounds that area is also worked out. So they enter the cells of the organ and resides in the cells for an unknown amount of time before leaving to move to other organ or to be excreted. So at what time you will inject it, up to what time it will stay to the different organ, and finally at what time they will eliminate from the body. So it causes a severe serious questions regarding this toxicity of the nanoparticle itself. And the toxic effect are what is the allergy. So it is a fibrosis, deposition of different organ, inflammation, cytotoxicity, tissue damage, ROS generation, and obviously that causes the DNA damage. And if you inject the particle in the rat and mice, it is very easy to observe the behavioral pattern of the mice and the rat, because the behavioral pattern of the rat and the mice itself helps us to analyze what are the nature, whether it is allergy. If it is allergic, causes allergic reaction, the mice of the rat always busy to itch their legs, their toe, their lip, their whole body. So that is a severe allergic reaction. Fibrosis, that is inside the lungs, deposition in different organs, so the time of urination, so time of feeding, and inflammation, whether any area is swelling, cytotoxicity, that means it can be analyzed by the trap and blue or any other method. Tissue damage, whether histologically you are able to analyze. I will show my next session slide, how the toxicity the damage can be controlled by the nanoparticle in different tissue level, and also the radioactive oxygens, free radical, and the DNA damage. So these are the toxic effect of the nanoparticle. And the toxicity depend upon the several factors. This is a very important point before one can prepare the nanoparticles for their experimental analysis. The nanoparticle properties that influence toxicity are the size, surface area, shape, aspect ratio, surface coating, crystallinity, dissolution, and any other factors. So I broadly pointed out some of the criteria: name of the chemical used for synthesis, whether it is gold or silver or titanium, types of precursors, concentration of the precursors, duration of the exposures, personal susceptibility, mode of entry, size of the nanoparticle, environmental factor, and threshold value. Threshold value in terms of the crude agent LD50. I'll explain one by one. Type of precursor, here we use the venom, right? Now, if you consider myself is during my PhD, I worked on the scorpion venom, Indian black scorpion Heterometrus bengalensis. The LD50 is 11 milligram per kg body weight, right? So 11 milligram, mind it. But during my postdoc after PhD work, so I worked with the scorpion venom, where the toxicity of the Vipera raceli is at 3 microgram per kg body weight, right? So microgram and scorpion is milligram. So it depends upon the agent. If anybody is like to tag the gold nanoparticle with the scorpion venom and gold particle with the scorpion venom, Vipera raceli, presently known as the W. raceli. So the types of precursor plays a very important role for this binding process, right? So concentration of the precursor, that means in terms of the LD50. So how much amount you bind? So either it is 1:1 or 1:2 or 2:1. So that dose you must be standardized, because this is very important, and all the standardizations can be worked out through the characterization of the nanoparticles, and also as well as the in vivo or histological observations of the animal in which you have injected the nanoparticle. So that toxicity of the nanoparticle, depending upon the, out of that, the surface area is a a very important size and surface area. As the nanoparticle size decreases, the ratio of the surface area to volume exponentially increases, which in turn increases biological and chemical activities. And when the size of the nanoparticle decreases, the number of the surface molecule expressed increases 50% fold. So the cytotoxicity of the nanomaterial results from the interaction between the nanomaterial surface and the cellular component. Thus, even when nanoparticles have the same chemical composition, they can have a significantly different level of cytotoxicity depending on the surface area and the particle size. In other words, the nanoparticle have a higher toxicity in comparison to the bigger particle with similar composition. So some scientists reported that the nanoparticle size dependent on the acute toxicity, what I have mentioned, and that too also differed with the strain of the mice. If you use the bulb C mice, the toxicity level may change. If you use the Wistar rat, the toxicity level, what we observed in the bulb C is totally different. So the strain of the mice is here also important, right? So some scientists make this observation. The particle toxicity depends size, but that too depend on the strain of the mice after intraperitoneal administration, because intraperitoneal administration, that means it is a very fast absorption, right? So histopathological changes such as the thymus cortex, apoptosis, focal necrosis, any other necrosis of the valid organ, that means liver, lung, kidney, that also being considered to assess the toxicity level. Even the cardiovascular toxicity can be mentioned by different scientists. That depends upon the nature of the nanoparticle size and the shape. In case of the administration of nanoparticle via inhalation, that means not through injection, it is a simple inhalation of the different size nanoparticle, showed specific distribution pattern in the respiratory tract. That means if you inhale the nanoparticle particles, the route and up to which the organ that particle reaches, that is also a very serious problem. And throughout the respiratory tract, they observed the accumulation of the nanoparticle in a different dose. So the toxicological evaluation of the response to inhaled nanoparticle requires the knowledge of the dose of the nanomaterial deposited in the respiratory tract. So if anyone is willing to work with this nanoparticle, that scientist or that group is very much cautious about its distribution and its toxicity inside the living body. So for that aspects, this nanotoxicological branch gains much attention to the scientist. And naturally, as because we are working in the venoms and the toxins, which is a very toxic, so this toxicological part plays a very significant area regarding this neutralization of the venom induced any changes of the living body. So even in the lungs or lungs damage, lungs histology plays a very significant attribute for these toxicological evaluations. Now, properties of the nanomaterial, what I have already discussed: novelty in both form and function, unique chemistry and physics by design. There are a lot of shape. It may be circular, it may be rhomboidal, it may be triangular, it may be cubic, it may be helical, prism-shaped, rod-shaped, hexagonal. But to be very honest, as because our team and I am indebted to my mentor, Professor Gomes, who is from the physiology department, and myself is a zoology, and all the scholars who are basically either physiology or zoology background, we have a very little knowledge regarding the chemical aspects of this nanomaterial, what we used So for the determination of the shape and the size of the nanomaterial, we have a collaboration, strong collaboration with the biochemistry department of University of Calcutta, Professor Anjan Dasgupta. He actually carried out this analysis, details analysis of the chemical part of this nanoparticle. So the typical shape of the nanomaterial, what I discussed, that is spherical, triangular, cubic, hexagonal, oval. There are a lot of rod shaped or prism shaped that can be analyzed by the chemistry person. I am not in a position to analyze how this analyzes, but the method, what they use for this analysis, that we know. But in true sense, how this interpretation can be done, if it is a iron code, if it is a silica code, if it is a gold code, how that analysis can be done, and that is carried out by the chemistry person. So the size and the shape is a very important factor. Shape, especially shape, is an important factor that play a vital role in determining the biological reactivity as well as toxicity. Right? This is very interesting, because if the shape change, the toxicity change. If the shape change, the biological reaction may be changed. The typical shape of the nanoparticles, I have already told you. The cellular uptake of the carbon nanomaterial or spherical shape and the tubes of the multi-graphite sheet was observed in the epithelial tissue of both gut and the gill in the fish. Right? One can easily analyze in the fish, because a lot of waste material are there in the aquatic system, either sea or the freshwater. So the fish is a good model for analysis for this accumulation of the nanoparticles, especially if you considered about the zebrafish, because it's a transparent fish. So you can start from the spawning to the adult life cycle. A lot of work have been already done and still carried out with the nanotoxicity in terms of the development of the fish, because it is easy to handle the zebrafish Danio reario. So because the whole body is transparent, you can easily observe the spinal cord defect or any other defect, the fin defect. So that in that case, the fish, the accumulation of that different shape and size can be observed in the gut and the gill. But in the gut and gill, the scientists cannot find the cube-shaped carbon nanoparticle. That I am trying to say. The nature of the nanoparticle is very much important, and in different organs, the different nanoparticle deposit takes place. Silver nanoparticle were found to be more harmful than silver nanosphere in zebrafish. The spherical nanoparticle are taken up in a greater number in cells compared to the other shape. So the circle or spherical is more biocompatible. Gold nanoparticle causes less accumulation of autophagism than gold nanosphere. So whenever a foreign particle enters into the body, so autophagism is there. So that is involvement of the autophagism here is a very less. And in truly and honestly, we have not worked out after the injection of the gold nanoparticle conjugated with the venom by Pararasili, what is the role of the autophagism? That area we have not ventured, but that is true. That area also open. So what are the in biological system, the different cell organelle behave? What are the condition of the mitochondria? What are the condition of the Golgi? Lysosome comes into the picture because autosome is there, is trying to engulf by the phagocytosis. But I do not know, because we have not worked out, and I hope or I think so that this area is still in a virgin. That area is not very common for the scientists to work out. So the spherical nanoparticle are taken up in a greater number, and the cytotoxicity property of the gold nanoparticle starts rod as well as spheres against human fetal osteoblast, osteosarcoma, pancreatic cell line, that has been also carried out in different human model. Maximum used gold particle is used. Another important factor is an aspects ratio. Right, I have not written in this slide here. The nanoparticle aspect ratio is the width to height ratio. What is the length and breadth? as an aspect ratio of 1 represent a spherical particle, while a nanotube has an aspect ratio close to 0. The greater the nanoparticle aspect ratio, the higher the toxicity of the nanoparticle. So if naturally for experimental and therapeutic uses, we have always tried to focus such a molecule and to keep the aspect ratio low so that the toxicity of the nanoparticle is also low. It causes the minimum alteration of the living body, right? So the aspect ratio is a very much important. And next point is a crystallinity structure. Crystalline structure may affect the toxicity. So the novelty in both form and function, unique chemistry, complex interaction with the biological and environmental milieu, biopersistence, that means how long it will stay inside the body, ready dispersibility, possible bioaccumulation, tissue penetration, irreversible biochemical and material activity. So these are the broad area and the properties of the nanomaterial mainly carried out by the chemistry group of people. So the characteristics of the nanomaterial that can give rise to toxicity, the particle size and shape that I have already discussed and already told, an important material characteristics. Size of the particle decreases, surface area increases. The change in the physiochemical and structural properties of the nanomaterial with a decrease in size could be responsible for a number of amount of material interaction that could lead to the toxicological effect, because or toxicity by any particle is not desirable, right? Because all of us will try to use the nanomaterial for the benefit or for the betterment of the any disorder. So I have used the nanomedicine. Here I have worked out to reduce the toxicity of the venom. So for the betterment of the nanoparticle, use of the nanoparticle, one should definitely play mind, definitely keep the concern about the property, about the surface area, about the aspects ratio, about the structural property, so that the minimum toxicity can be achieved in any biological system. So what is the reason for this toxicity? The surface area to volume ratio of the particle, which increases their interaction with the surrounding molecule. This is one of the very justification line, why they have given the toxic behavior. Chemical composition of the particle which is responsible for its reactivity. Say for gold itself is a toxic. Surface change of the particle is responsible for electrostatic interaction. So there is an chemical reactions or electrostatic bonding changes, and the complementary of the nanostructure could cause inhibition of the enzyme activity, either competitive or non-competitive. So any involvement of the enzyme definitely is there, because in my earlier slide I have already mentioned that the ROS is involved. Reactive oxygen species is very much important. And for the ROS analysis, there are a lot of enzymes, glutathione, phosphatase group of enzyme. Lot of enzymes have been involved for this ROS analysis. So we are not still clear whether inhibition of enzyme activity takes place by either in competitive mode or non-competitive mode, and that causes the toxicity. We are not, in case of the venoms and toxins, it's not clear to us also. And the disease associated with the nanoparticle exposure, just look at a glance. I have already, sorry if the spelling mistake is there, is the mitochondria, nucleus, cytoplasm, membrane, lipid vesicle. Lot of lipid vesicle, that means autophagosome. Lot of area has to work out, that comes to the brain, that is neurological disorder, Parkinson's, Alzheimer's. Nanoparticle inhalation, it comes to the lungs, it causes asthma, bronchitis, emphysema, cancer, circulatory atherosclerosis, vasoconstriction, thrombus, high blood pressure, heart arrhythmia, heart disease, death, and other organ unknown etiology. We still do not know what are the level of the damage. And the another is autoimmune disease. Autoimmune disease, dermatitis, urticaria, vasculititis, Crohn's disease, and colon cancer. Now, here I am must explain some of the interesting area that I observed during my research. So, in the last phase of my career, I worked out with the black tea, right? So, suddenly I got a paper that tea and coffee causes toxicity in the human being, and excessive consumption of coffee and tea causes preeclampsia, right, in the pregnant mother. So that line causes a very curiosity to me. So immediately after an extensive study with this tea, I submit a project and worked out with the tea toxicity. So anyone, if I discuss about the tea toxicity, whenever I told the tea toxicity, the people are very much surprised that tea we consume daily, at least 2, 3, 4 cups per day. But that tea is causes the toxicity if we consume in the pregnant mother, right? Now come to this area. This is a disease of unknown etiology in a kidney and the liver. Why I am concentrating that point? After consumption of the tea in the pregnant rat, in early pregnancy rat, I just carried out the simply histological analysis of the kidney and the lungs. I found that the tea in a high dose that causes the disruption of the Bowman's capsule, right? That I have proved, and the paper I have already published. So there is still a conception whether tea nanoparticle causes any damage to human being, especially for the pregnant mother. So what material we consume daily, that particle may causes some changes in our body. So the disease associated to nanoparticle exposure, that means this chart is incomplete. Still we do not know at which area, in which organ they will cause the damage, and there are exposure is millions. I just considered about tea, but in our daily routine, if you think which particle causes the damage internally in your organ in the form of the nanoparticle, we do not know. So the nanoparticle internalized in the cell is a very dangerous. And now the scientists are trying to understand the mechanism of this Parkinson and Alzheimer's, whether this aspect, whether any nanoparticle deposition in the amygdala regions of the brain. So it is still debatable, but that point should be taken care of this toxicological assessment. And nanoparticle induced oxidative stress is a very emerging area, because oxidative stress is among the commonly reported stress that nanoparticle induce following exposure on a cellular level. Oxidative stress can be broadly defined as a lack of balance between antioxidant activity and the production of oxidants. A state of oxidative stress arises via increase in the ROS production favored over antioxidant are generally produced in the form of the byproduct of biochemical reaction, including neutrophil mediated phagocytosis, enzymatic metabolism of cytochrome P450, that is a biotransformation pathway, and mitochondrial respiration. So the ROS attack nucleic acid, protein, lipid, most vital biomolecules which can lead to NADPH like system, activation, electron transport chain, membrane depolarization, damage to mitochondrial structure, anything. So the sphere of the damage of this ROS is tremendous. So our target to reduce the ROS at any cost so that it cannot causes the toxicity. So nanomaterial of different substances and their toxicity, that is metal substance, non-metal substance, and polymeric material. Metal and non-metal are commonly used. we have not used the polymeric material, so I do not know what are the toxicity in terms of venoms and toxin, mind it. So polymeric material, how they will react inside the living biological system, we do not have any idea. We have worked out only the metal, more particularly the gold and the silver. So the metallic substances is aluminum, gold, copper, silver, zinc, iron, titanium. But I have an experience with only the gold and the silver, and non metallic substances, carbon based nanoparticle extensively used, silica, and the polymeric substances is there. And the nanoparticle, look, the aluminum oxide, gold and copper oxide, they have an itself toxicity. Now aluminum is also very serious because you know the aluminum toxicity causes the Alzheimer's disease. It is the main cause of the aluminum inhalation. And for this reason, in our house, we often restrict the use of the utensils made up of aluminum. So the aluminum itself is a toxic, and it is a cytotoxic, also causes the genotoxic, disturb the cell viability, alter mitochondrial function, increase oxidative stress. Gold, it is cytotoxicity of the gold. NP are dependent on the type of toxicity assay, cell line, physical and chemical properties. This line is important, and remember, I request all the participants to remember that line, because whenever we use the gold nanoparticle, that causes the cytotoxicity. Now, the gold nanoparticle tagged venom, if we use this as an anti-cancer model, right, the venom tagged gold nanoparticle in a leukemic cell line, we interestingly found the loss and the mortality rate of the leukemic cell line is vigorous. So one can suggest that this venom tagged gold nanoparticle can be used to treat the cancer of different cancer. We worked on this specifically the leukemic cell line, breast cancer cell line, where we found a terrible and good result. I will show you that slides and what are the data with relation to W. russellii or Naja cautia. We worked on the two specimens. One is Chandrabora, another is a Gokro or Keute, whatever the maybe. So different species though, the Naja and Naja Hana. So the tagging of the gold particle with the venoms that can causes a severe decrease or severe mortality of the cancer cell line, especially the leukemic cell line. So the application of the gold is a very itself beneficial. Then copper oxide, toxic effect on the liver and the kidney, genotoxic and the cytotoxic, along with the disturbing cell membrane integrity and the oxidative stress. So this is a metallic substances. And nanoparticulate toxicity, zinc, these nanoparticles have been accounted for change in the cell morphology, DNA damage, alteration in the mitochondrial activity, human hepatocyte and the embryonic kidney cells. And iron oxide, these nanoparticles bioaccumulate in the liver and other reticuloendothelial systems organ. And magnetic iron oxide, nanoparticle bioaccumulate in the liver, spleen, lung, brain, barrier, and inhalation. So there are a lot of disadvantage of the other particle. In that case, gold is rather more safe, right? Titanium oxide may possess a genotoxicity because there is a very alarming genotoxicity. And the lung inflammation, they also toxic effect on immune function, liver, kidney, spleen, myocardium, glucose, lipid homeostasis. And the last one is a silver, more toxic in terms of cell viability, generate reactive oxygen species, and the lactate dehydrogenase leakage. Lactate dehydrogenase leakage is there, so naturally that had effect in the electron transport chain, mitochondrial biosynthesis pathway, etc., etc. But the serious problem with this radioactive reactive oxygen species, right? So whenever we use the silver gold nanoparticle, one should very cautious about the ROS generation. So there some method you must simultaneously apply it for this decrease of this ROS activity. So the nanoparticle of non-metallic substances, that is carbon-based nano material that is extensively used in computer, electronics, or any other area. So it poses site-dependent toxicity. Multi-walled carbon nanotube have produced carcinogenic effect. Single-walled carbon nanotube in the liver causes disturbance, and the silica causes generation of the raw subsequent oxidative stress, also biochemical parameter along with the hepatotoxic effect, so that causes the problem in the liver. Now this is a very interesting slide, that is a nanotechnology in Hollywood. That means it this line I have mentioned, it is a fantastic voice, mainly that has been first worked out and comes into the picture by the NASA, right? So NASA, what NASA actually performed, if you write in the Google the fantastic voice and the NASA, you can find several video, YouTube video is there. What they are trying, they can use the small vesicles or vehicles that is known as the nanobots. That nanobots are introduced in the astronaut's body, that can reduces the radioactive exposure in the space, because astronaut can gain much of the radioactive exposure from the space. So to reduce this radioactive exposure, they use the nanobots that can absorb, instead of damaging the internal body tissue, they absorb this radioactive material, and the astronaut will be saved. So there comes in the Hollywood came out with a movie. Based on that, a movie has been prepared in 1966 that provided the public with a glimpse of the future of the nanoscience, because this is the future, obviously, and depicts a miniature surgical team that was injected into a man to operate the blood clot on his brain. I have told you the astronaut, but here this slide show that not the open surgery. The surgery is done by the nanobots. What the doctors usually introduce these nanobots into the victims, and naturally that will dissolute the clot. That idea nowadays is trying to use to reduce the mortality against the snake bite poisoning, because in India snake bite poisoning causes a various serious health issue, especially in our West Bengal, in Sundarban area. A lot of people died due to lack of treatment, because antiserum is not available. Though antiserum is available, but that the preservation process is not ideal, because in many of the village there is no electricity. So the local health center, they do not have the refrigerator, so they do not have the antiserum to preserve that. So the antiserum are present in the district hospital. So in Sundarban, the patient brings back to Canning Hospital or Shagordigi Hospital. So the duration of the venom inside the victim creates a very serious problem, because since the birth, the concept of nanotechnology has been employed in various sectors, including information and computing technology, energy. But in the venom research, that the application of the nano technology is a very rare. So we think that the application of the nanoparticles or the noble property of the material has opened the door in future to diverse application that that will bridge this distance problem and also save the life of the victim, right? And so this concept, this surgical team, the nanobots, creates us a good hope, at least in the case of the snake venom poisoning. Now it is in nanopharmacology, that is also a new area. So the drug, if I explain in a very lucid way, say for example, there are 4%, A, B, C, D, right? 4% is there, and 4% is affected with a fever, high fever. So immediately what we usually carried out, we take a paracetamol, right? So someone take paracetamol as a crocin, someone take paracetamol as a calpol, someone take the paracetamol as a UGC plus, and someone take paracetamol in other form of the drug, right? But my question is that, or the question asked by the scientist, which drug is more better for a person? Crocin, Calpol, UGC plus, or other. So whether the absorption of the drug is same in all the person, whether the activity and the action of the drug is same in all the person, no. So for that reason, in abroad, usually the doctors carried out the pharmacogenomic study, right? That is already started in India in AIMS. Once the doctors suggest a medicine, he will test your blood which drug is very much suited for your phenotypic conditions, right? Say for antibiotic A is suitable for me, but that antibiotic is not suitable for you. So a new area has opened in the medical science, what we called as a pharmacogenomics, and more precisely nanopharmacology, because pharmacology is the area where the drug can be chosen. So a new branch pharmacology that is rapidly emerging. So being a zoologist, I have some interest in the pharmacology because my supervisor was a doctor, and he is MD in pharmacology. And still I remember my first day in the laboratory, sir came and gave me a book, the title Pharmacology by some author, right? And that pharmacology is a pure textbooks of a medical science. So I just surprised, but later on I understand, because our lab is working with the venoms and toxin lead to therapeutic drug, right? So by the creations of the drug, formulation of the drug in that early time, that is not a question of nano science. Early in that period, the Goodman and the Gilman, name of the author, that book is still in the medical service, medical education. Whenever you choose pharmacology, that is a textbook. So that area is intimately associated with this toxicology. Why? Because whether gold is best suitable or titanium is best suitable, that you have to work out, right? For venom, whether gold is good or silica is a good, or silver is a good, so you have to standardize. So that is a pharmacology. It is the application of the nanotechnology to the development and the discovery of the drug delivery methods. And target the drug delivery is a diagnostically and therapeutically. Diagnostic means it is a detection, and therapeutically means it's for the wellness, right? So this is a nanopharmacology. And what are the sharp difference between conventional therapy and the nanopharmacology? Conventional therapy, decreased bioavailability. Nanopharmacology, protection of molecule from metabolism and degradation. It is a wide and non-specific distribution. It is able to reach specific target site. Increased systemic toxicity, that is reduced systemic toxicity. Increased risk and benefit ratio, risk benefit ratio still unknown, we do not know. Drug resistance, there is a lower and infrequent dosing. Still we do not know which dose is very much compatible to reduce the toxicity of the living being. Whenever I try to tag with the nanoparticles, that portion is very much clear when I'll show you my work with this nanoparticle and the snake venoms. So the lower and infrequent dosing or risk or benefit ratio, that is also very much negative aspects of this nano particle and its toxicological aspects. So ultimate goal, nanomedicine is the application of nanotechnology to achieve breakthrough in the healthcare, enables early detection and prevention of the disease, a new area treatment and therapy, overall improved diagnosis and treatment, comprehensive monitoring, control, repair of all human biological systems, and molecules at molecular level using engineered device and nanostructure to achieve the medical benefit. So the blue line is actually the take home message. So it causes the early detection, it for the better treatment and therapy, and it for the molecular level, it for the engineering device for the particles, nanostructures, and obviously last of all the medical benefit is obviously there. So the gold particle, there are some pros and cons of the gold particles. Why we are prefer, the scientists prefer the gold particles. So the gold plays a very important role in the living system. Just explaining nanomedicine, then I am come to gold. So nanomedicine, it is an interdisciplinary field of science. It is a chemistry, nanotechnology, biology, and application means it is a diagnostic, that means imaging, identification, therapeutic, that means delivery of medication to exact location, killing bacteria, virus, and cancer cell, that I have already mentioned, the cancer cell, and repair of the damaged tissue. Also, it can repair the damaged tissue, right? So other diagnostic application in the market, colloidal gold particle, rapid test for the pregnancy and ovulation. These are the some new area where this gold nanoparticle can be used. But that gold nanoparticle, I have used ARA for the venoms and the toxins, but there are several area of the use of the gold nanoparticle, say for the biomedical imaging. So that is a prospect area, is somehow related with the physics also. This biomedical imaging, magnetic nanoparticles, cell sorting and clinical diagnosis, and silicon nanoparticle diagnostic imaging, super paramagnetic iron oxide, that is MRI, and magnetic iron oxide nanoparticle detection of Alzheimer's plaque, what I have already described, to locate the amygdala regions. Right. So one thing I have forgot to discuss, that regarding the genotoxicity of the nanoparticle, that is also a very interesting area, but I am unable to do that, the genotoxicity. The mechanism behind the nanoparticle associated genotoxicity is majorly due to the overproduction of ROS species, and which in result increase of the oxidative stress too. Hence, oxidative damage to the genetic material also. The nanoparticle mediated production of ROS can be due to intrinsic production interaction with the cell target or inflammatory reaction. The resultant damage to the genetic material can be direct or indirect, primary clustergenic or secondary. The primary toxicity occurs due to the interaction of the nanoparticle themselves with the DNA, whereas in the secondary genotoxicity, the genetic damage occurs as a result of ROS. In indirect primary clustergenic mechanism, exocyclic DNA adduct are produced via unsaturated aldehyde produced as a result of ROS. So naturally, the genotoxicity area will be well analyzed, and it is much deeper, considering the alteration of the DNA, also considering the alteration of the ROS element, and scientists have worked out in different with different cell line, and also in the gold and the silver, they causes the genotoxicity changes along with the neurotoxicity changes. So the applications of this nanoparticle is a very important. And now coming to the gold, why gold gives a special stress? Now, gold nanoparticle are arguably the first nanoparticle that are used in commercial material and approved by FDA. Now, though we are the independent country, but regarding the drug, we are still helpless, because a lot of spurious drugs are available in our market throughout India. But in that case, FDA is a very strong, the Food and Drug Administration of USA. So that agency first approved the application of the gold as a nanoparticle. So that is first use. And or nanomedicine, that can be used as a nanomedicine or nanocarrier. Moreover, these nanostructure poses a unique size-dependent surface plasmoresonance property, what I mentioned as an aspect ratio, that made them utilizable in biosensor application. Lot of biosensor mechanism has been applied in our daily life, say for daily sugar detections, etc., etc. So the biosensor application, this gold particle can be extensively used. That's approved by this FDA based on this. And then in recent times, several studies reported different factors that can lead to cytotoxicity towards the viable cell. And the size-dependent toxicity of the gold nanoparticle towards the human embryonic stem cell and their neuronal derivatives is less compared to other particles. So naturally, all the scientists who are specifically working naturally নবনীতা মাইক বন্ধ নবনীতা মাইক বন্ধ
Hello.
So all the শুনতে পাচ্ছি ঠিক আছে So
Okay, please continue.
colloidal gold particle, just a minute. So the gold particle, in that case, gold particle creates a very interesting, right? So other diagnostic approach, therapeutics, these are the new area. Drugs delivery form under the investigation, work has already formed. That means inside the nanoparticles, if we use the dendrimer, if we use the nanoshell, that is already I have mentioned in the astronauts. Liposome is a very important artificial biomembrane, because in the rural village, the people are still very afraid for taking injection of any important drug, say for antibiotic. But whether that drug can be easily available with the nanoform with the help of the liposome, that is artificial biomembrane, work is going on. The micelles, insoluble fat material, quantum dots, and nanocrystal, these are the area where extensively work has been worked out in the nanomedicine for the drug delivery system. So my question is that whether we can use the dendrimer or the nanoshell or the liposome, whether the venoms, whether the antiserum can be available in the form of the liposome, whether the antiserum can be available as a quantum dots and the nanocrystal mode. So these are the new area of the nanotoxicology where we can able to address. Next, what I just explained very short the what is dendrimer. So it is a man-made molecules, tree-like structures, but man-made molecules have a tremendous disadvantage. I have already mentioned, right? So you just look at the pictures, the drugs and the targeting ligands, and this is the imaging agent. So man-made molecules, tree-like structure, many small branching molecules, application, cancer cell recognition, diagnosis of cancer, drug delivery, reporting drug level in tumors, reporting cancer cell death. Nanoshells, core of silica with a metallic outer layer, usually gold, again gold, linked to antibodies that recognize the tumor cell. So once the cancer cell take up the nanoshell, an infrared light is applied and taken up by the nanoshell. Intense heat is created which selectively kill the tumor cell and not the neighboring healthy cell. That is a very upcoming methods. By using the nanocell, scientists are trying to kill the cancer cell in vivo. So if it is possible, then there is no questions of radiotherapy or chemotherapy like this. So they create the nanoshells with the nanoparticle. They can tag with the ligands, tagged with the healthy cells, and that healthy cells infrared light can be applied. That will destroy the cancer cells, but without damaging the other normal cells. So this is a nanocell concept. This is a nano drug. This is the first drug developed by the USA, not in India. So amphotericin B, the liposomal injection, amphotericin LS. So this is intravenous injection, doxil, nano D3, vitamin D3, because you know nowadays the vitamin D3 the decrease of vitamin D3 causes a serious problem both in the ladies and also in the child, especially in the child. And newborn baby is often given with the vitamin D3 injection, the lack of vitamin D3. So that can be already in the market. The nano drug, the doxorubicin is a well-used antibiotic, methotrexate, flubiprane, lot of drugs. Paclitaxel, that is an anti-cancer, amikacin, amphotericin B, and also the vitamin D3. That is a very common. If you have a search with the Google, you find the vitamin D3 and the nano particle. You find a lot of articles with there that nanoparticle tagged with the vitamin D to reduce the deficiency of the vitamin D3. Now, what is a broad difference? Nanoparticle application and toxicity. Silica, platelet aggregation and reproductive toxicity. Ceramic, cancer drug delivery, oxidative stress, cytotoxic activity. Super paramagnetic iron, MRI contrast, oxidative stress. Oxide nanoparticle, disturbance in iron homeostasis. Gold shell, hepatic and splenic toxicity. No generation of ROS. This is very advantage. Titanium, central nervous system. Silver, endoplasmic reticulum and stress. And carbon nanoparticle, pulmonary toxicity and intestinal inflammation. What scientists are trying, if you use the other nanoparticle, they will try to reduce all the toxicity level. So there are methods by which you can reduce the platelet aggregation. If the platelet aggregate, then there is a problem in the blood clotting system, right? So scientists are now trying, along with the nanoparticles, some agents have been tagged that can inhibit the platelet aggregation method, but still in a pipeline, not in a final form. So that toxicity can be checked or reduced by any form. What are the advantage? Reduced degree of invasiveness, reduced systemic side effect, cost effectiveness of the medical and surgical therapy, benefits of cancer therapy, reduced morbidity, but disadvantages are there. Lack of proper knowledge about nanoparticle effect on biochemical pathway, not only the biochemical, the in vivo system toxicity, different effects of the drug due to particle size, and difficulty in the ADR monitoring. So monitoring means patient to patient, it can vary. So there is not huge data. What are the variation level in which person A and person B, in which point they are differed. So that data is still missing. Molecular mechanism, it causes oxidative stress, it causes inflammations. That means the nanoparticle induces apoptosis, nanoparticle induced DNA damage, nanoparticle induced ROS study that can easily be carried out. Inflammation, there are a lot of pharmacological method by which one can easily analyze the inflammation method, because in normal inflammation, that means it is a swelling of any part, either internally or externally. So external inflammation can be created by the injection of the yeast carrageenan. If you inject the carrageenan in the rat paw, immediately within 15 minutes, the rat paw will swell. That is causes inflammation. Now, my question is that if you inject the gold nanoparticle after this inflammation, whether that can also address the inflammation procedure. So one can easily address the inflammation, accumulation of the nanoparticles, whether it can accumulate, so biotransformation procedure, toxic effect of a particular organ system, counting of MFO, mixed function oxidase system, glutathione, etc., etc. So one can, that can be addressed. This is the picture of the nano robots. This is they are now ready for this. Nanosurgery is there. You can often find a advertisement, knee replacement surgery by robotic surgery, right? So here it is the small devices is known as nanobots or nanorobots, detect and repair the targeted damage and infections, biomedical instrumentation and nanosurgery. These are the upcoming line of the nanomedicine, but still not in application in the human. Is a future vision, and I hope that within a few years we can be able to use these nano robots in our to combat our health disorder. Nanobots is an emerging field technology creating a machine or robot perform tasks of a nanoscale dimension, size, বলেই দিয়েছিলাম naturally the 10 to the power minus 9. Nanobots are made up of a DNA of a bacteriophage. So it's just a vehicle. So also called nanorobots, nanomachine, nanomites, and nanoids. Nanoids is a very common term. Nanorobots are tiny machine used to cure the disease in human. They need large numbers of nanobots to perform well in macroscopic and microscopic tasks. In future, medical nanotechnology is expected to employ nanobots injected into the patient to perform work at a cellular level. Different type of nanobots are already standardized and in experimental trial. One is named as a respirocytes. It works in artificial erythrocyte. It must have a more oxygen and natural RBC. It is very useful for patients suffering from anemia, sickle cell anemia, thalassemia, etc., etc. Microbivores act as an artificial WBC, also called nanobatic phagocyte. It absorbs and digests the bacteria in the bloodstream. Plotocytes, it acts as an artificial platelet. It reduces the time for blood clotting. It may allow the complete hemostatic in little as 1 second, less than 1 second, even in a large wound. So if it is a large wound, so people are the defective in blood clotting factor, so you use the clotocytes nanobots that can easily and within less than 1 second that can able to clot in that wound site. So these are all in an experimental trial. This is a future application of nanomedicine. So nanorobots in respirocytes treatment, that respirocytes has been extensively worked out, and I think within a few years that can be applicable to human being. Treatment of anemic condition, cardiovascular, neurovascular disease, treatment of tumor, diagnostic, fetal abnormalities, child-related disorder, respiratory tract breathing disorder, underwater breathing, transfusion of blood, and perfusion of organs. So comparison between the natural RBC and the respirocytes. So this one is a nanobots, right? So you just find they are biconcave, disc-shaped, no nucleus. These are spherical in shape, made up of diamonds. Lifespan 120 to 5 to 8 months. This is only one type of RBC found in the blood. There are various sizes depending upon the rotor presence inside it. Cardiovascular system. 4.5 million normal level at a 10 to 12 million. It supplied 4 oxygen molecule. It carries more than 200 molecule oxygen. Hemoglobin, rotor sensor, microchip detectors. No hemoglobin is there. Produced in red bone marrow, artificially made. They move in the blood vessel, eventually squeezing the capillary. They move in the blood vessel normally in a specified path using flow of blood. A very low RBC count will result anemia, no side effect. RBC do not have nuclei in human, has a microchip to monitor. Round and can change, have different kind of shape depending on the number of shorting rotors used in keys in what RPM that can be rotated. That is a criteria for the different types of respirocyte. Different shape and size of nanobots are being produced by the doctors. These are the nanobots, rough pictures, glycoprotein-based structures entered into the human blood, so meaning the intravenous vaccinations. So there is a small pump is there, so that is called rotor. So that rotor can actually control the entire movement, is known as a capacitor. So that can, and also the camera through which the doctors can visualize whether that robot will directly act correctly or in a wrong way. So these are the diagrammatic representation: single-stranded bacteriophage DNA, short triple-stranded, and these are the triple-stranded hexagonal barrel have been produced, and aptamer lock. Aptamer is a regions actually is a separate area, the aptamer. And in a recent time, this aptamer plays a very important role, and in nanotechnology or more specifically the nanoscience, these are plays a very important role. It is nothing but a short single-stranded DNA or RNA molecule that can bind to a specific target, such as a protein, with a high affinity specificity, acting like a chemical antibody. This is nothing but a chemical antibody, right? They are used in diagnostic and therapeutic and have advantage over antibodies, including greater stability, easy of modification, smaller size, allowing them to be used in the drug delivery and clinical applications. So these are the artificially made instead of normal antibody. Initially, I have already mentioned that in the Indian Institute of Science, Bangalore, the scientists, a group of young scientists are trying to replace anti-serum against snake bite. They use the small nanobots. These are the aptamer-based vaccine technology or liposome-based vaccine technology. I think within 2 or 3 years, instead of anti-serum, that application of these nanobots through aptamer will be available. So aptamer is a very short single-stranded DNA and RNA molecule that can bind to a specific target, such as a protein molecule, with a high affinity and acting like a chemical antibody instead of normal antibody what we observed. So antigen unlock aptamer and exposed payload trigger, because there is a small capacitor there that will actually trigger in which directions these nanobots will act, in what level they will act. So these are the some pictures. Nanobots usually killing the virus in the human body. Nanobot used to detect the pathogens in the blood. Nanobots cured the kidney stone. So not available in Indian scenario right now, but the experimental trial has been already successful. So the nanorobots induced, and that nanorobots has a little toxicity, mind it. So the scientists are also perform that experiment to reduce the toxicity of the nanobots. So aptamers, these are the definition what I have given. This small ligand, and the technology what they use is known as a SELEX technology. That means systemic evolution of ligand by exponential enrichment. Aptamer are produced by in vitro selection. These all are synthetic nanobots or nanoparticles. So this aptamer, using the aptamer in nanoscience or nanotechnology, is a very upcoming area through which the different therapeutic aspects can be resolved. So it can check and repair DNA. It used to clear the coronary artery blockage. It can also cure the problem in spinal cord. It can able to detect the stenosed blood vessel. Nanobots are put into the act in purifying the severe waste to reservoir in the field of the environment. So environmental clearance, in that case, the nanobots are also used. So there are a lot of use. Look at a glance. Arteriosclerosis, breaking up kidney stone, prevent diabetes, gene therapy, gout, drug delivery, fight against cancer, breaking up the blood clot. So advantage, no operation, cost surgery low, less risk, faster, more precise diagnosis, speed up the medical treatment. Drawback, replication may become out of control, right? Complicated maintenance, এবং ওই the capacitor which is present inside the nanorobot may fail. So that nanobots may act as an unguided missile. And initially it is a very cost initial installation. Nanobot should be accurate, otherwise harmful effect. So that is the major disadvantage. If it is uncontrolled, if the capacitor is out of control, then this nanorobot may damage the normal tissue as well. So hard to interface the customer and the designing. So the nanobots are used in medicine, are predicted to provide a wealth. When the severe side effect of the existing therapies are considered, the nanorobots are found to be more innovative, supportive to the treatment and diagnostic of the vital disease. I think within a short period of time, all the disadvantage can be overcome, and that can be extensively used in therapeutics. Now, this is my last slide. Now, I have finished here with this slide. In the next class, I start from here. This is the latest article, 16 December 2024, the scientific reports. Nanobody-based recombinant antivenom for cobra, mamba, and wrinkles bite. This work actually carried out in the African regions, because the mamba bite is a very common. There's a cobra, that means is a spitting cobra is there, and wrinkles is another venomous snake in the African regions. So there, the work has already been started to produce the nanobody based recombinant antivenom. Conventional antivenom therapy is gradually diminishing. Lot of papers have been already published that nanotechnology and considering the nanotoxicology application of the nanotechnology in the snake venom research is inevitable. So we hope that within and future these nanobots or nanobody based antivenom treatment therapy can be available in our country. Thank you.
So thank you very much, Dr. Dashgupta. Now the session is open for few discussion and questions. Here are already participants put some questions.
Yes.
One question from Arijit Das. He asks that biodelivered, you have used biodelivered nanoparticles for venom binding. Are they capable of reducing the toxicity of the venom like synthetic nanoparticles?
Yes, it is absolutely reduces the toxicity. And in my next lecture, I will show you. All of us will know this the plant, common plant, Rauwolfia serpentinea, Sarpogandha, right? Initially, in Ayurvedic science, that is mentioned that this Rauwolfia serpentina possesses an anti-snake venom activity. So initially what we have carried out, we make an extract from the plant, right? And that extract is used in the venom-induced mice. We observed that the mice is survived. So there is no effect of the venom. So what is the
Fantastic.
conclusion? Conclusion is that the plant contains some material which neutralizes the toxicity of the venom. In the next phase, what we have done, we took this plant extract tagged with a gold nanoparticle and then again inject into it in the mice model. We observed the toxicity level, that means fold of protection, is much higher in case of the nanoparticle induced gold particle. So definitely that is visible, that is
Exactly.
more applicable.
Exactly. Thank you, sir. Another question from Dr. Shambhutu Chatterjee. I think if there is any method to target into particles specifically to a tissue. Actually, he wants to ask that during ordinary drug delivery, they has to face the blood-brain barrier, but whether this nano-based technology could overcome this blood-brain barrier.
Yes, absolutely. That can overcome blood-brain barrier, and there are so many.
Please unmute, sir.
Yes, that is a very interesting part. In the next class, I will discuss how we can measure whether this nanoparticle is more effective, more size, that it will not cross the blood-brain barrier, and more effective to neutralize the toxicity.
Exactly. Dr. Rajoshi Ghosh has a question. Could nanobots eventually replace certain immune functions, acting as artificial phagocytes?
Yes, a possibility is there to replace the immune function, because these are all artificially controlled. But the main question is that if the capacitor will be out of control, that then nano robots will be a villain that will causes the danger to normal cell as well within a second. So the scientists are still in a dilemma whether the nanorobots control will be from the outside through any chip, or it can be maintained internally. So the question is very pertinent. Yes, it can alter the immune system as well.
Exactly. So if anybody has no question, then I have this two question.
Yes.
You have I known from your lecture that there are respirocytes as artificially made, and in our body, the normal RBC has its specific shape and size and diameters for oxygen carrying oxygen. So is there respiratocytes can able to maintain the demand of oxygen?
The table
Yeah,
what
because yes, because among the mammalian group we know the demand of oxygen varies from species to species. So this must be very much species specific respiratocytes has to be made.
Right. So the scientists are now trying to prepare the respiratocytes considering the human only, right? If you consider
Exactly.
the other animal, that obviously vary because the lifespan, the length and the diameter of the RBC, oxygen carrying capacity is absolutely different from species to species. So
Exactly.
considering human, what information I have gathered or till now that is available is considering absolutely on the human, but it is not flawless. That I am
Exactly.
to say.
Exactly. And another question is, you have told that it induces apoptosis also. So how could this artificial nanobot technology discriminate the normal cells and the cancer cells, so that if it uh, hampers the normal cell cycle or regulations of the
The
P53 gene?
vehicle has been used through aptamer, and through
Okay.
vehicle the human DNA is imprinted, so they can identify the specific target site. So the
Okay.
aptamer is a very upcoming area.
Okay.
So that is a ligand binding, just antigen antibody binding in that way.
Okay, okay. Thank you, sir. And Anita Gande and Dr. Kaushik Sharkar has raised hands. So first I approach Anita Gande. Please put your question.
Sir, good afternoon, sir. Sir, I am working in the field of horticulture, sir. In nanorobotics and nanoparticles, the farmer will not accept that one, sir, in our Karnataka side. Can we use in all these in farmer fields, sir, in vivo and in vitro also?
Yeah, that is a very interesting area, but very honestly, I do not have any idea. But you may explore the application of the nanorobots in the agriculture or floriculture, whether that will help to kill all the external parasite and also help to the plant the hormone which are very helpful for this blossoming, right? Like florigen, the activity of the florigen, so or
Okay, sir.
auxin or cytokinin. So I do not have any ready information to me, so you can search, and obviously it is a good area to explore.
Okay, sir. Thank you, sir. Thank you.
Okay. Now I approach Dr. Kaushik Sharkar.
Thank you, ma'am. Thank you, sir. Actually, I have get the answer from previous question. Okay, no problem. No.
Okay, okay. Dr. Anisha Chaudhary, please.
Thank you, sir, for your very nice and informative lecture. I have a general query. Sir, nowadays we have a tendency to drink water kept in copper vessel for long time. How much is it safe for regular consumption? As you, sir, you have told about metal toxicity. That's why this
It is a
question.
very good and pertinent questions. I advise not to drink the water which is present in the copper bottle.
Okay, sir. Okay, okay.
Prefer the glass bottle. The glass bottle is more safe. In my house, all this container has been replaced by the glass bottle.
Okay, okay, sir. Thank you so much. Actually, I do drink water from copper vessel. That's why this question came
No,
to my mind.
you do not know. Then the copper particle
Yes.
in the form of the nano particle that may arises in your drinking water and you consume. You do not know, and that effect can be
Yes.
visualized after a few years.
Okay, okay. It can generate toxicity in the long
Yes,
term, long term
absolutely.
use.
I suggest you not to drink.
Okay, okay. Thank you, sir. Thank you so much. Okay.
Okay. Dr. Shoumova Chatterjee, perhaps you have a question.
Sir, thank you for this illuminating lecture. I have a query that you have told about nanobots. My query is how nanobots will sense its target. That is, how it will detect the defective gene, leaving the correct one, or how will detect its correct location, leaving
Yes,
the
yes.
proper other one, and will target
When I show my slide, you notice that there is a DNA aptamer. So the DNA and the vehicle is the DNA or RNA. So the target tissue DNA is there. That is the source, and the binding is nothing but an antigen antibody. Is a chemical binding. but the binding is simply antigen antibody. How the antibody will identify the antigen inside your body. Say for malaria vaccine, that is already out but not available in India. So malarial parasite inside your tissue, either RBC or in the liver, right? So whenever you administer the malaria vaccine in your body, how the antibody will identify the malaria parasite inside the RBC or in the liver? Basically the same method.
Antibody interaction, sir.
Yes, this is known as a chemical bonding through aptamer.
Okay. And thus also it will actually determine which gene to repair and which one not.
Yes, this is a preliminary trial. I think more work has been gone. If you check through Google or PubMed, the use of aptamer or use of nanobots or respiricides, whatever, you get plenty of papers. But all this research are still in pending because the scientists are failed to address the adverse effect of these nanobots, right?
Okay.
Nanobots, exactly. There needs much more research in this area. Okay, so I think Dr. Dashgupta has satisfied all of you, and thank you, sir, for being present here and spent your valuable times. And I'm really thankful to you because you have focused on the new area of research in combating various diseases, including cancers, and also focused on new terminology like nanopharmacology, pharmacogenomic study, etc. And so thank you all, and thank you, Dr. Dashgupta, again. We may contact you whenever we have required you. Thank you, sir.
Thank you.
Okay, sir.